appears to us as a single flash is not a single flash at all. There's lots of different modulations in that particular flash. And it is possible in a scientific paper, for example, to depict that flash. So here's the intensity, and here's the intensity of the flash, the duration of that flash, what we call the interflash, the in between the flashes interval and intensity. And you'll notice that they're all, even these ones are looking a little bit different. The life cycle of a firefly is everything. It's about basically everything that happens from when an egg is laid until we see the adult. And fireflies have a life cycle that's pretty much the same as moths and butterflies and wasps. There are 4 stages in this life cycle. The egg hatches into a larva. Larvae molt. They shed their skin so that they can actually grow. It allows them to grow. And then they transform into a pupa, and the pupa transforms into an adult. And the important thing, and I can't stress this enough, is that the larva is the longest stage in the life cycle. So in our Australian fireflies, they may well have a 2-year life cycle, and 2 years is a long time to spend in a life cycle. And most of that will be spent as the larva, and a very, very short time at the very end of it is when they emerge as the adults. And I'd draw your attention to these pictures here. The pictures I'm going to show you are pictures of fireflies that occur in India. This one is Abscondita terminalis, and here is the male on the top right. You'll notice that you can see the very large, light organ that the male has, and also the very large eyes. And I'd like you to contrast that with figure E, which is the female. Her head and the eyes are much smaller. It's one of the ways that we can often pick a female straight away. But she has much less light. She can be picky. She can be fussy. She doesn't have to produce so much light. And her light comes from just one segment in the abdomen, and there are a couple of segments after it. Whereas this one, the light organ, is the end of the abdomen. Here are the larvae, upside, dorsal and ventral, and this is what the pupa looks like. Fireflies, remember, fireflies spend more time as a larva than anything else. And whatever affects the larva will affect the life cycle. If the larvae can't survive, the life cycle doesn't survive. We need to know where they live. And it's interesting, we're not really good with our terminology here, because we will talk about terrestrial insects, and that's okay. That's a good term. Terrestrial insects, if they're terrestrial fireflies, then both their adults and larvae are on land. But when we talk about aquatic fireflies, what we mean really is that the adults are still free flying. The adults are still terrestrial insects. It's the larvae which are the aquatic forms. And there's a couple of different sorts of larvae which are aquatic. And this particular one, Dr. Barua might recognize this, because this species is on the campus of your university there. This is the genus and species is Sclerotia substriata. And these larvae are very, very hard-bodied. They're quite small. None of these things are much over about 10 millimeters long. And where they swim just under the surface of the water, just under the surface of the water. They take in atmospheric oxygen. Although they're aquatic, they actually need to breathe in atmospheric oxygen, which they do by means of I've gone one too far. By means of the spiracles, which are here. And the only way that they can get those spiracles to pierce through the water is to swim upside down, and they actually push those spiracles through the water. The other alternative, of course, is this is a quite different type of larva. And I'm not sure whether this particular type of larva actually lives in India. And I am going to be saying this a lot, and I make no apologies for saying it. We don't know, I'm not sure, things like that. That's good. That's good, because it just means that there's that much more for other people to find out. However, if you look at this, these are the legs, 1, 2, 3 pairs of legs. And this particular larva, which is very, very soft-bodied, has gills down the side of the body. So it has to take in dissolved oxygen, not atmospheric oxygen. And the other thing about this one, whereas this one is as tough as old boots, I would not like to be a predator trying to eat this particular type of larva, whereas this one is very soft, probably moves around on the floor of the pool, swims sometimes, maybe just moves slightly, very, very susceptible, were it not for the fact that in association with those gills, it has very, very distasteful glands that it can extrude, and they are not very nice for anything that's trying to eat it. This is another species. It's, again, this one, you'll notice from the label even that this one says it's from Bengal. This is Asymmetricata circumdata. This is another common species in India, and very pretty looking species too. And it's terrestrial. So both the adults and the larvae this time, but this is a different looking larva entirely. And you'll notice that what happens with this one, let's look at this, which is the underside of the larva. Here's the main body of the larva here, and the plates of the larva are expanded at the side. Now, if you know what a pie dish beetle is like, tenebrionid, then this is the same sort of effect, in that with those plates being expanded, it's sort of sealed down, so that it's another mechanism of protection for it. Now, it would be nice to know just what is actually in India, and I know that later on Dr. Gupta is going to talk to you about it, but I want to tell you how you might go about finding that out. And the logical starting point is that you start with a catalogue. Now, the catalogue I'm going to recommend is nearly 60 years old, and it's this one. It's McDermott. We always just refer to it as McDermott 1966, and it's a catalogue of the world fauna. This is important to realise, that this is fireflies of the world. It's not just the fireflies in India. And it's organised to reflect the scheme of classification too, that makes it easier for us to use. But we have to sort of spend just a tiny little bit of time looking at what that scheme of classification is. Now, remember, I did say for if you're an entomologist already, you know all this. I don't have to tell you this. I'm not talking to people who know all about it. I'm talking to people who might want to know about it. And what we do is we organise things. It's a bit like going into a store. You go into a store and they have organised the groceries so that it's easier for you to access them. You find similar things in similar places. And here we have a whole system of classification that we'll just look at the way it is for the fireflies. Fireflies, we've already established that they belong to a class, the class which is the insects. They belong to an order within that class, which is the Coleoptera, and we've looked at how those endings sometimes will tell you what level of classification you're looking at. They belong in a family, Lampyridae, which is, again, the ending tells you that that's a family. And here we start to get to some more sub-classifications. Subfamily, there's a subfamily, the Luciolinae. This is the one we're going to be talking more about than anything else, and it ends in INAE. And then a genus and a species, which is the name we gave to the firefly from the previous slide, which is Asymmetricata circumdata. Now, I'm sorry, but we have to just spend a little bit more time looking at something like this, which is not all that interesting. But McDermott, when he did his classification, was the first to admit that it was not a very natural classification. And he tried very hard, although in what he was doing when he developed this 1966 classification was he was following what had happened before. And the catalogue that happened before him was developed by a Frenchman, and it was developed in 1910. So that a lot of these subfamilies had already been developed by 1910. But what McDermott did, and he was very much aware that it was not very easy to work out which subfamily you were actually even in, he tried to go, particularly in this subfamily. Notice that they all have AE on the end, so these are all subfamilies down this row here. McDermott developed a whole lot of further subdivisions, which he called tribes. They end in INI. We're getting more complicated. And he was even able, with some of them, to take them down to the level of subtribes. Now, McDermott was the first to admit, again, that these were not natural classifications, but they were just convenient classifications. And it's still handy to be able to use this to go back and try and find it. Now, if we have a look over here, there's a little bit happened in this intervening period here between 1966 and now. Various people like Roy Croson in 1972, or our Russian friend Kazantsev in 2006 have come up with other systems of classification. And that's not all of them. But it is important, I think, that if you are going to be a student of fireflies, that you are aware of the fact that the very first, the most recent classification is this of Gavin Martin and his colleagues in 2019. And let's have a look at how it compares, because it compares very favorably. Whereas McDermott gives you a lot more information, you still need to be aware when if you are going to actually be talking about fireflies, that there is a most recent classification. But let's look at this one, the Amiditini. Martin thinks that it should be a subfamily too. But within the Amiditini, McDermott had this as a subtribe, and Martin put it up to the level of its own subfamily. Within the Luciolini, McDermott had a tribe, Ottotritini, and Martin put it up to its own subfamily level. This one is going to go the other way, because McDermott never ever dealt with a particular genus of fireflies, which I haven't spelt here for you, which I must apologize for. But the genus is called Pristolicus, and Pristolicus is the most beautiful looking thing. It's pink and it has lots of black markings on it. Sensational looking firefly, except that it doesn't have any light organs. So it was not, in McDermott's system, included in the fireflies, whereas a lot of people now have done analyses which indicate that it should be included in the fireflies. So it's one that is going back towards the left. The rest of the classification is pretty much the way McDermott had it, except that, of course, there's down the bottom here, the Siphonocerini and the I can't even say this word properly, I'll give up. But these are new subfamilies since McDermott. But I put those two in green because it's important. Right at this moment, you're thinking, oh, Leslie, my head is spinning. These are the only two subfamilies in India. They're the only two subfamilies in India. I will mention something about this later on, but at the moment, these are the only ones we know about. So, and let's have a look now that we've done that, let's have a look and see what McDermott looks like. So if we look at, let's come down here on the first page, subfamily. You'll notice that it's subfamilia, which means it's in Latin, but you can read that. That means subfamily. Here's the Luciolini, and he's divided into a tribe. So the very first tribe is the Luciolini. And while I was stressing that there are ways, you do eventually get to remember these things for yourself. but there are ways to remember these things because they have different endings to the words. There's also different ways of pronouncing them. And this pronunciation was taught to me by Jim Lloyd. So this is Luciolini, but Luciolini. Luciolini, Luciolini. You probably won't remember that. And down underneath that, there's a genus Luciola. And McDermott went even further. And if you look up at the top of the next page, he decided that there should be some subgenera, that is just a subdivision of the genus in Luciola. Now, the problem with Luciola and the subgenus Luciola is that there are over 270 species in it. And there is no way that it is all the same. They're all the same genus or subgenus, and we'll talk about that later. But running down the left-hand side here of page 99 are all the species in alphabetical order. And associated with each species is quite often quite a large amount of references to literature, particularly, let's have a look at these two because these two are from India. And Acera was described first by Gorham. Gorham was a minister of religion. Apparently ministers of religion had a lot of time, spare time on their hands in the early days. And here's the only thing about this now in looking at these references, and these are the sorts of references that you have to access ultimately, is that we would now be required in a scientific paper to spell these references out in full. It was not that way 60 years ago, and it was simply considered appropriate that they should all be abbreviated like this. So this catalogue, even though I'm going to indicate to you that there may be better ways of going about it, there's still a lot of information. You still need to be able to go back to this catalogue and look at the things that we can say that within the fireflies, there's a subfamily Luciolini. Leslie Ballantyne works on the Luciolini, which is why she's going to be stressing it so much. This subfamily is divided into tribes. This was how McDermott actually classified fireflies when he did his catalogue. Within the first tribe, the Luciolini, and looking under the genus Luciola, subgenus Luciola, we find there are going to be many species from India. I just showed you one page. There are 270 odd more species. They're not all from India, but there are lots of pages in this catalogue. The first species we saw in this catalogue was Luciola acera. This is how we would write it, just in case you're wondering that this is the genus. The subgenus goes in brackets, and the name acera was followed immediately by the name of Gorham, because Gorham was the man who first described it. Now, where do they all come from and where do they all fit? Well, let's try to do a process of elimination, really, because they say that there are no fireflies in the polar ice caps. So, and I haven't got the polar ice caps in my map here at any rate. There are no fireflies, funnily enough, in this eastern part of the Pacific Ocean. So some of they simply don't extend further east from Australia beyond about Fiji. So Hawaii, Samoa, Tahiti, places like that, funnily enough, don't have fireflies. The fireflies there are fireflies in Canada, but the fireflies of the United States, Mexico, and South America have all been, most of them, the predominant subfamily here is the Lamperini. They've been extremely well worked on. From our point of view, at what's in India, these fireflies do not occur in our particular area. And I want you to have a look at this because this is rather interesting. And I may be able to I might be right. I hope I'm right in that the area I've indicated on here is the area that I work on. I started off in Australia and then I just gradually expanded. And when I expanded, I realized that for a start, this is actually quite a natural area. There are natural boundaries. There are deserts and mountain ranges all sort of around here and ocean around the rest of it. If you go over to here, that there's a little piece just over here somewhere where that's New Zealand. New Zealand did have a species of firefly recorded from it, but it's incorrect that there never ever was anything in there. But why have I rung it round? Not only because it is, it's a natural area, because the species of Luciolini only, the green subfamily that occur in this area, do not occur anywhere else. So that's quite a useful thing. Here's the area. So as far as, and here I go again, as far as I am aware, as far as I can pronounce, these species do not occur anywhere else. Now let's go back again, because we haven't dealt with this, Africa. There are more than 90 species of Luciolini in Africa. And the problem is that nobody has worked on them. So we don't know. There is some work starting just recently on fireflies from the island of Madagascar, and which could be really, really interesting to see. But we have no idea about these species here, whether they are there. We don't think that they're the same. We don't think that the genera are the same, but we don't know. As for the rest of it, there are Luciolini through Europe, but there's not a lot of different species. There's only a few, actually. So like Luciola italica, Luciola lusitanica, but fortunately, again, people are starting to work on them now, and it may well be that there's a whole lot more species than we think. If we look back to here for a minute, just in case you wonder exactly where I am, that's where I am at the moment, about there. And in case you were wondering, yes, it's midwinter here. Now, the reason that I kept hedging my bets a little bit about catalogues and McDermott is still a useful starting point. It's a useful starting point because it is a catalogue of the world. This is like another catalogue, but it's not a catalogue, it's a scientific paper. And it's a scientific paper about the Luciolini of Southeast Asia and the Australo-Pacific region. I'm the first author of it, and it is dealing specifically with all of the genera. If I can talk about that particular area, then I'm talking about all the genera, with the exception of one that occur within the that particular subfamily. This paper actually gives you all the information you need to work on the Luciolini. Now, why do I stress this? I stress it because I've worked on it, but I also, as you will see later, stress it because this amount of work has not been done on other groups. So I'm not saying, look, I'd like you to work on the Luciolini. I'm just saying that if you want to work on the Luciolini, there's a lot of information to help you. It will contain a key to genera in the Luciolini. So it contains, once you have keyed your specimen to genus, then it contains a profile of each genus, and what you'd normally do is you would go to the profiles and you'd say, okay, I think that my specimen might fit in there, and then you'd see whether it did, or what do you have to do. This is the really useful part of this paper. And if you look across, this is a list of the species. And what I did was I picked out specifically the genus Abscondita, because I know that occurs in India. And if you look at it, here are some localities within India and Southeast Asia that where the genus Abscondita occurs. You'll notice that this is a column which tells you where the type specimens are. I might talk a little bit more about types later, or somebody might want to have a question about it. This first one, these are all abbreviations, obviously, but that's Paris Museum. This is the Forest Research Institute of Malaysia, which is where Dr. Nada is from. This one, the one below that, is the Civic Museum in Genoa. For him again, this is in Brussels, and this is the Natural History Museum in London. And you'll notice also that whether there are females associated, this is quite important to know the nature of the female, because not all females are flighted. And if they can't fly, then you immediately have a problem with the distribution of your species. So do they have females? Are there larvae? And is there anything from India? Yes, there is. There's this species. It's now called Abscondita perplexa, and it was described by Walker in 1858, a long time ago. But it also occurs in Sri Lanka. So Abscondita perplexa, we know that this one's right. We're not 100% sure about this record, as you'll see in a little while. The paper that I was talking about also has a key to species and a profile of each species. And at this stage, if you aren't already wondering it, you might say, well, why did you spend so much time on McDermott's catalogue? And the whole thing about the McDermott's catalogue is that it's only the Luciolini that has this established taxonomic framework. If you want to work on anything else, you have to go back and do all the work I had to do to get to that particular point. Now, this is quite important, because this is the other two subfamilies that the Lamperini that occur in, remember I said there was only Luciolini and Lamperini that occurred in India, but within the Lamperini, these are some of the genera that occur. And once again, you may have a bit of trouble with my accent, but from the top, there's Diaphanes, there's Pyrocelia, which used to be known as Lichnurus, and Lamprigera, which used to be known as Lamprophorus. Now, Lamprophorus tenebrosus or Lamprigera tenebrosa are the two names for your biggest, most famous glowworm. The Indian glowworm is the most amazing thing. I have actually seen specimens of it, but they were dead when I saw them. But I think this is a wonderful thing to see. Now, these are the genera that if you were to work on the Lamperini, and these are bigger, I would encourage work on these groups, because they're much, much bigger than the Luciolini. The Luciolini get to be about 10 millimeters long, whereas these ones are considerably larger than that, much, much easier to handle with. Now, I put in this other subfamily as well here, called the Amiditini, because there's another genus that belongs in there called Vesta. Vesta is a lot like Diaphanes. So Vesta and Diaphanes look very much alike. And Diaphanes, I'm not sure whether you understand what it is if a piece of material is referred to as diaphanous, it's semi-transparent. And Diaphanes has, on its pronotum, it has a couple of very transparent areas on it. And I presume that's where the name came from. Now, the problem with Vesta is that not even Martin in that most recent catalogue could decide where Vesta should go. We don't know. We can't define it properly. People keep getting confused about it. So it's been put in the too hard basket. The second part about this particular slide, though, is that you may think that work on, there hasn't been any work on Indian fireflies. There's been a lot of work on Indian fireflies. It's just that it appears, the people I've listed here, probably from about the 1920s to the 1960s, were working on fireflies. There appears then to have been a bit of a lull. And now we do actually still have, we have people now. You have people in India working on fireflies, and I think most of them are working on Luciolini. It's easier sometimes to work on a group that has an established framework. But these people, most of these are Indian anyway. And this is my favorite person, J. Samuel Raj. He did lots and lots of work, and he worked on Lamprophorus, the Indian glowworm. So lots of them worked on Lamprophorus. I've given some of these their up-to-date name, but a lot of them worked on this one, which is Luciola gorami, which must, we can only assume, since it keeps cropping up all over the place, must have been very common. And these people worked on Lamprophorus as a mechanism of control. So there was a lot happening. There has been a lot happening in firefly work in India. There was a bit of a lull, but it started again. There are people working on fireflies in India. Now, I've been giving out hits right, left, and centre, but I imagine that when we get to the question centre, some people might suggest what this might be, but there's a sting in the tail. It's not as obvious as you think. Now, there's a lot of there's a lot of information here, and there's heaps more information than I'm ever going to be able to give you in the period of this talk. But I'm going to refer you to a website. Now, this is my website. I set it up specifically with students, Firefly students in mind. So there's a heap of stuff there. One of the things is access to literature. That the literature will, of course, be literature that I've written, but each paper will have a large amount of references at the end of it, so that you can get exposed to a lot of the literature you need. It's ease of access for you. And while we're at it, I'm sure Dr. Barua will be looking at this later, but these pupae, which I've got here, were from a colleague of mine, Jinhua Fu, in China. And you'll notice that while this, I think that this is the male and this is the female, I'm only going by the size of the head. The male head is a little bit bigger, but you'll notice that they're luminescent all through the body. It's not just that fireflies are necessarily able to produce light from the end of the abdomen, and we don't know why. Now, let's say that you say, thank you very much, Dr. Ballantyne, for your talk, but I'd really like to try to identify my specimens another way. And that's perfectly all right. But there are drawbacks. You want to go to a museum. And that's where you run into trouble. In Southeast Asia, you run into trouble with museums because the early history of these countries, in terms of for you in India, it's the British. For elsewhere, it could be the French or the early explorers who came over, the Dutch, the people who were collecting early on in your country. Took the fireflies, they took them back to Europe or to Britain, where they were described, and they were put in museums, and the museums are in Europe or in Britain. So in some respects, it's like your fauna is not really your fauna at all, with these types being overseas. It's a consequence of our shared history in that we are in the same sort of situation to some extent in Australia. So when you go to the museum, you're not going to see types there, and you should be very critical of the identification of material in that museum. Now, let's say that you do it a slightly easier way. You find an expert. You have got in touch with somebody, and that somebody said to you, yes, you may send me, I'll identify some specimens for you. And you run into a real problem. Government regulations will not allow you to send specimens out of India. So Sriram Murali, wherever you are, if you're watching right now, you know that this problem has happened too. And I hope that you managed to identify your monsoon firefly, for example. You can tell me later if you did. So what do you have to send? If you cannot send a specimen, then you are getting a second-class identification. And even if it's me, and I'm the one that does most of these identifications, you are getting a second-class identification. So photos, if you were to send me some specimens and I identified them from photos, then you should at least do me the courtesy of saying that they were identified from photographs and not from actual specimens. Or if you do it by yourself, so number 3, you want to try by yourself and you've decided, I'm not going to work on the Luciolini, I want to work on Diaphanes or Pyrocelia, which is fine. Well, you have to build up a taxonomic coverage like I did with that other paper. And that is not easy. It's quite a difficult business to do. Okay, now you might think, well, okay, what else is left for you to do? Everything is left for you to do. I mean, I haven't told you very much at all, actually. What have I actually shown you here, really? I've shown you a classification scheme. Sorry, we go back one. Our classification scheme for the Luciolini. I showed you an overview of the genera and species that occur in a very specific area that includes India. I've shown you some descriptions and some pictures. That's just a start. The rest of it's Dr. Gupta is going to be talking very specifically about your fauna. Everything else is waiting for you. We need to know all sorts of things. The life cycle. We don't know this in Australia, and life cycles are often quite difficult to do. How long is the interval from egg to adult? How many species do you actually have? How many genera? Is it possible that you have more than we are telling you? Are there really only the 2 subfamilies here, the Luciolini and the Lamperini? What sort of environments are they in? How do we protect those environments? Dr. Sharma, when he was giving the introduction, was talking about a firefly park. Now, that's a way around. I've said here, is firefly tourism feasible? But I wonder whether it is. Why does the monsoon firefly synchronize its flashes? Well, it's a mating dance, isn't it? But how does it synchronize its flashes? Do you know? And what species? Sriram Murali is the monsoon firefly. It's important that you know that we are a community and that we know that firefly researchers know each other. So today you've met me. Now you will meet Dr. Nada. You'll meet Damika Wijikun from Sri Lanka and Sarah Lewis from the USA. We know each other. We don't just know each other because we see each other on television, on this sort of platform. We know each other because we meet each other at conferences. So this is a really useful thing. We have a Facebook page, and it's most. I suggest that you join it. This is how you start to become part of a community. This is how you start to find people who will help you. And this is my wish for you. I was going to put the Firefly poem from Where the Crawdads Sing, but I thought I might get into trouble there. If you could possibly have part of the passion that I felt for this research, then I would be giving you it would be my pleasure. I think that's the end of my talk, and now I wonder whether we have some questions.
Professor Ballantyne, and any questions from anyone? They are welcome to ask questions to Professor Ballantyne. Any questions, please?
Here we go.
Madam, one of the persons, Arun, says he can't see fireflies anymore in his village. Any specific reason? He can't see fireflies. Earlier, when he was young, he used to see. Now, he is not able to see fireflies in his village.
Well, the trouble is what's happened to the habitat. Where were they before? Have they cut down the trees? Have they altered the habitat somehow? Or there's a problem of light pollution with fireflies as well, that the if there's too much light, then the fireflies just can't compete with light. Light is actually pollution as far as fireflies are concerned. But it's mostly, it's very likely that it's been some disruption of the natural habitat for the firefly. And did anybody try to identify that specimen?
Not yet. We are just at it, and maybe we will be able to identify this specimen soon.
Okay.
Madam, another enthusiast asked that the fireflies have been put in superfamily Elateroidea, and
Yes.
we also have click beetles. Some of the beetle taxonomies think that the fireflies may be separated from the superfamily Elateroidea. What is your opinion, madam?
Well, they are, the Lampyridae is part of the Elateroidea, yeah. But there are luminescent species in the Elateroidea as well. So, there's not just elaterids like click beetles or Lycidae, the family Lycidae as well, but it would appear that the Lampyrids are quite possibly a reasonable sort of distinct family. But I'd suggest that he goes and reads Martin's 2019 paper, actually.
Okay. So we know that fireflies communicate through light or bioluminescence. Do fireflies communicate chemically? Is there chemical?
Yes. Yeah. The thing is that not all fireflies have light. So we have things like the Pristolicus, that pink, the one I told you that was pink with the black markings on it. I know of a species that I've described from New Caledonia, which doesn't have a light organ. So that yes, they may do that. And what you would expect to start to see would be if they're relying on pheromones, then you would expect to see development of the antennae. Most of the antennae of the fireflies that I've been talking about are quite short because they're relying on the light, so that the thing is more large eyes. Whereas if you're starting to rely on pheromones, eyes aren't so important and antennae become a lot more important.
Yes. Yeah. Madam, one more question is there, that fireflies larvae are carnivorous. So larvae of fireflies are carnivorous. Is it a general statement, or are there any exceptions?
I'm not aware of any. They do have their mandibles, their jaws are pierced by a canal, so they can grab hold of the prey. They inject salivary juices into the prey, which tends to stun it or to paralyze it. And then they can digestive enzymes in to liquefy it, and they can suck it all up again, sort of thing. So I would think, given that they have such specialized mandibles, then I would not think that there would be any alternative, unless, of course, they're maybe just sucking up a little bit of plant juices occasionally. But we just don't know. This is another one of those things that we don't know.
Yeah. There is one more enthusiast, Ms. Shivangi Singh. So she asks whether firefly eggs also have the property of bioluminescence.
Yes, they do. and that's one of the odd things that, I showed you those beautiful pupae, which were luminescent. And they probably live in a little hole in the soil somewhere. And you would wonder quite what why they might be luminescent at that stage. But it might be a protective, it might be an aposematic sort of display or something like that.
Okay. Okay. There is also one more question from Rahul Pradhan. He asks the question as to why fireflies are important in nature, and does it come under any specific food chain or web?
What eats fireflies, in other words?
Yeah.
Well, given, I would imagine birds, lizards, toads, all sorts of things will eat them. But the problem with, in some cases, if they have defensive mechanisms, like lots of fireflies are very bad tasting. So, but no, they have a place. There'll always be something which will eat fireflies. Yeah.
Yeah. There is one more question. Is database, that is world checklist of fireflies, available? And if it is available, is it accessible? The world checklist of fireflies, is it available? If it is available, is it accessible to all?
Well, there is no world checklist. There's McDermott's catalogue.
Okay.
Yeah, or there's not yet, because we just don't have enough people working on fireflies. So
okay.
yeah.
Yeah. Yeah. Any other questions from the participants? Yeah. One of the questions, again, a youngster asks, geometric morphometrics, can it be used as a tool in firefly identification?
I wonder who that might be. It's not somebody from Thailand, is it? It may be able to be. I've only seen one paper where it's been used, and they already knew what they were measuring. And one of the problems with it is that if you can imagine that this is, can you see that, Parvez? You can see that all right? Yeah, okay. Yes. Here's the front part of the body. This is the pronotum. It always does that. It droops. So when you are trying to do this measurement of the whole body, you need that up like that, not like that. And so I think it'd be really nice, because it'd save people having to go through all the complicated taxonomy that we have to go through. Yeah,
correct. Now, this Dr. Abhijit, he's asked one more question. The weaver birds, the granivorous birds, they use these fireflies to light their nests inside. So what they do is, during night, they pick up these fireflies and they put it in their nest, which is a well-woven purse. So what comments you have on this? The birds picking up the fireflies to light their houses.
I hadn't heard that before. That sounds like a remarkably adventurous occurrence. No, I can't. I don't have a comment.
Yeah. There's one more question. They ask, in conclusion, fireflies, are they beneficial? Are they harmful? Are they neutral?
Well, it depends on who's doing the looking. That there was extract of firefly has been used in all sorts of areas, like in the medical areas. It was used when they first sent up the spaceships, when we were first exploring space, to see as a bioassay. For me, I just love the thing. So of course I think they're beneficial. They're a tourism for more than anything else at the moment, in terms of trying to benefit communities. They could be a very important tourist enterprise.
Yeah. There is one more question via iPhone, and the questionnaire is asking, since the larvae, they spend a lot of time in soil, is there any relationship between the soil health and larval development?
Well, remember what the larvae are doing on or in the soil is they are living on snails and slugs and very small invertebrates. So I can't, apart from the fact that the soil, it's a rather a complicated question. It's quite possible that yes, it could be, but I don't have enough information.
Okay. Any more questions from participants?
There seem to be a lot of questions here on the chat, on my chat thing.
Yes,
but
yes.
Can I ask a question?
Coming out? Yeah, please.
Hello, ma'am. Thank you for the talk. I'm wondering, like, the adults that emerge in every season, right? Like, do their numbers vary across the years in a natural pattern? Like, for example, in this year, if I see some 1,000 fireflies in a particular locality, and then the next year I go back to the same locality to kind of get a count of the adults that emerge, and if the count is coming down, could we say that their population is declining, or is there a natural kind of a pattern in their
you'd have to do observations over a much longer period than that, and you'd need for you probably have a lot of rain over there. I live in Australia, the driest continent on earth. So one of the things that we would notice was our populations will vary from year to year, depending to a lot of extent on the rainfall that we've had. So the rainfall is affecting the larval prey. It's affecting the snails and slugs that they might be able to eat. So there would be lots of other things involved. It has to be a much longer term evaluation than just over 1 year.
Thank you so much. So just a follow-up quick question, like if we are to monitor their populations, if we want to kind of find out if their populations are on a decline or not, what would be a good way to go about? I mean, I understand it has to be a long-term study for one, but other than that, how can we monitor them, would you say?
Well, you're going to have to try to find some way of doing a count on them. I think I'd save that question for Dhammika Wijikun, who is going to be speaking straight after me, and he's done surveys in Sri Lanka on just this problem. So that it may well be that he will give you a much better answer than I can.
Yeah.
Thank you very much.
There's one question from Nishant Venugopal. He asked the question, can we consider firefly a bioindicator for healthy environment? Bioindicator?
Yes, you can. Yes. Well, when you think about it, what do the larvae feed on? What will maintain the larval prey? It's all connected. Yes, they're a good bioindicator, actually.
Yes. So, madam, I think we're having a queue for you. There are a lot of questions more. I think we may move on to the next speaker, and definitely we will be able to answer the questions of participants at a later stage, as the questions are just trickling in. And we very much are thankful to you and grateful to you for having shared such a valuable information, the lifelong experience with us. And definitely we will be in touch in course of our studies on fireflies. Thank you once again on behalf of all the participants, speakers, and invitees, and especially the institution, EMPRI. Thank you so much, madam, and we look forward to meeting you again. Yes, thank you. Yeah. So, ladies and gentlemen, we move on to the next speaker from Sri Lanka, Dhammika Chandana. He is a lecturer currently serving in the Faculty of Science, University of Ruhuna, Sri Lanka, and has put in 14 years of extensive experience in identification and taxonomy of fireflies in Sri Lanka. He is also interested in birds, animal physiology, and wildlife photography. Dr. Chandana is a member of the Firefly IUCN Specialist Group and member of IUCN Firefly Program for funding. He was very kind enough when requested to share his work and experience on fireflies today. So, Dr. Chandana Damika, please.
Okay, can you hear me clearly?
Yes, yes, you are
Okay,
clear.
thank you very much, Professor Chakravarti, for your nice introduction and inviting me for this important event. I think you have organized this event for the first time in India, especially on fireflies. No? So I
Yes,
congratulate you and your team for future success also.
yeah.
So today my talk is based on fireflies, their significance and their status in Sri Lanka. Uh,
Yeah.
Actually, before starting my talk, I would like to thank my supervisors, Professor Hemanta Vegiriya and Professor Chitra Bogavath. They were the initiatives or the beginners of the firefly research in Sri Lanka in I think late 2009. They also introduced me to this field. And however, unfortunately, currently they are not in the country; they live in abroad. I grateful them first and Dr. Valentine. I wish my sincere gratitude to her for her invaluable support for identification of fireflies in Sri Lanka since 2009. Thank you, madam. Uh, Now I am moving to the next slide. Actually, uh, my general question is, how much do you really know about fireflies? I think I heard several questions in previous session. Some of participants asked that what is the beneficial or what is the significance of fireflies? No, I think I can start this from my childhood. Can you remember? Some of you also have some experiences. In my childhood, I enjoyed fireflies in my home garden with my parents and collecting them to glass bottles. Likewise, so you also have some experience. It is kind of one of significance of fireflies. No, that's why I select this topic because my understanding is as South Asians, we know very little about their significances. So before starting your research, I think as especially young researchers, students, or excuse me, uh, or any other scientist, you should know about their significances first. No, that's why I select this topic. My talk today based on these uh, subtopics: significance of fireflies on their tourism, the agriculture, medicine, research, and aesthetic value. And as a second step, I'm going to move to the status of fireflies in Sri Lanka, little bit about their past literature and the current ongoing studies and our future plans. Okay, I think this is a very common introduction. As you know, we commonly known as fireflies as a luminescent insect or the lightning bugs, but they are not bugs. They have tail light, and you know they produce this light as a result of the enzymatic reaction. But I am not going to talk on this topic because I think our next speaker will talk about this, the biology of bioluminescence. But I want to here mention that their light producing is a coolest reaction because it has happened inside the cells. It is somewhat different than the artificial light. No? Then I am moving next slide. Actually, this is, I think all of you know the body structure of the insect and also the fireflies. But here I want to highlight that some of features are important when you are trying to identify them morphologically in the field because you can see here their pronotum, the shape and also the light patterns of the pronotum are also important. for the identification and the dorsum color pattern, especially elytra and the structure or the actually the structure of the elytra is important, especially elytral apices. Actually, especially in South Asian fireflies, when you are studying fireflies in the field, some of actually you may suffer, actually you may face some problems when identifying them, because most of us, there are several species of fireflies in South Asia with orange dorsum with black apices. I think Dr. Valentine also agree with me. And when moving to the ventral side, actually their abdominal segments are important. Their body door ventral color or the abdominal segments color also important. However, you know the visible segments are vary according to the different genus, but this is Luciola. You can see here abdominal segment 4, 5, and 6 and 7 also represent the light organ. You know the shape of the light organ also important of the male when trying to identify them, and also the color patterns of the abdominal segment 4 and 5 also important. These are the some important aspects when you are trying to identify them morphologically. However, you want to follow the standard taxonomy keys for proper identification, and also you have to dissect these light organ and extract their adhesives and adhesive sheet for confirm their species. And not at all actually I think I am advise you please get the advice from the experts before confirm your species. That is important. Okay, then I am moving to the first important part of my talk, significance of the fireflies. Uh, First one is tourism value. You know the fireflies are one of important subject for the nighttime tourism. But unfortunately, I think in our countries, especially in South Asian countries, I think we are in some steps back comparing with the other Southeast Asian countries, because they have developed firefly tourism in some extent than us, because we are actually in Sri Lanka, we have initiated some aspects or some steps taken forward to develop the firefly based tourism in Sri Lanka, but you can also develop firefly gardens, river banks, night zoos, firefly trails, and firefly forest also. Uh, this is one of the important aspect you can generate the income. I think I don't know the situation of India. However, in Sri Lanka, you know, we somewhat depend on the tourism industry. So therefore, we are planning to develop the firefly-based tourism. However, we have enough infrastructures and the facilities to data and tourism, but we not yet some actually we are initiating some step to develop the firefly based tourism. It is kind of one important value of the fireflies. And you know, I think some you have heard that these synchronizing behavior of the fireflies. This is one of examples. This Potinus carolinus that lives in Great Smoky Mountains in North America. They have ability to synchronize the other flashes of the other firefly species or the sometime nearby flashes. So thousands of tourists visit there to watch them, to enjoy them, because the ultimate thing is they emit their light together, or they switch on and off together. That is a kind of fascinating behavior. So you can see, however, I think not yet recorded species in here. And I am moving to the next slide. Because this is also important as a young researchers and young firefly lovers. You can study their flashing patterns also. Do you know their flashing patterns are species specific. So these are some examples actually I taken from the internet, but you can see here their flashes are different, and even flashes in between males and females also different. So you can see here the different flashes with different time intervals. So these are the important aspect when you are planning to develop your ecotourism or the firefly based tourism in your area. Actually, I am really happy to heard that in several months ago, one of researcher in India have sent me an email regarding they have identified some areas about the for firefly tourism. Good thing. And I am moving to the next important value of the fireflies. They have agricultural value because some of I think some of one of researcher in your area have visited or they have tested it. uh, I also tested using in-lab experiments about the feeding efficacy of the firefly larvae and their utility as a pest control agent to control the snails and slugs. We know actually the larvae and also the flightless females. You know there are some species of fireflies. They have flightless females. These females and larvae are kind of voracious feeders on the molluscus, especially snails and slugs. But when I am talking about the Sri Lankan situation, we have large number of snails and slugs. They are invasive to the country. So we tested how to control these invasive slugs and snails using these firefly larvae, especially the larvae of the Lampigera tenebrosa. Lampigera tenebrosa. I heard that actually Dr. Ballantyne also mentioned that this species is common in India. Their origin also India. We have good evidence that base 1956 mentioned that in 1954 and 55, several hundred larvae of fireflies in the Lampigera tenebrosa collected and shipped to Hawaii, Indonesia, Philippines from Ceylon for the purpose of biological control of giant African snail. The scientific name of giant African snail is, you know, the Lysocatina fulica. This species is still common in Sri Lanka as a major pest of the vegetables, especially nursery stage of vegetables. Uh, Therefore, these are some links for future researchers or the young researchers. You can investigate them further. And I am moving to the next important significance on medicine. Uh, Scientists believe that these luciferin and luciferase of the enzyme and the surface of this enzymatic reaction of the bioluminescent are two rare chemicals. Scientists have tested these chemicals for the therapeutic purposes of diseases such as cancer, multiple sclerosis, cystic fibrosis, and heart diseases, and also muscular dystrophy. So they use these chemical as a indicator or the indicator chemical or the sometime uh, to test what is happening inside the body, especially changes of their cells. They can detect using these chemicals. So these are the applications in the medicine field. Now I would like to move to the next part. It is a kind of emerging trends of the application of fireflies bioluminescence. So this is a research value. You know the luciferase system is widely used in genetic engineering field as a reporter gene. Now scientists have encoded this gene to animals and also the plants. You can see here the they have tested or they have encoded these gene to laboratory animals, especially rats, mouse, guinea pigs, rabbits. You know the when it is important to precise the results. That means you can observe you are testing uh, animals at night without switch on the light by using these bioluminescent. So it is important to get the good results. So these kind of things now scientists have developed, and not only that, now there are some pet animals, cats, dogs also with bioluminescence. So now you can actually in recent future you can enjoy with your pets with the bioluminescence. And also several years ago, some scientists have encoded the bioluminescent gene to the tomato plant. Now it has developed to encode this to the uh, ornamental plants and the home garden plants. So now actually in recent future you can enjoy with your home garden with bioluminescent ornamental plants. So that these are the new research areas you can initiate or you can implement or you can develop further. These are the applications of the Firefly bioluminescent. And now I am moving to the next slide. It is also the applications of the research of the luciferin and luciferase. Some scientists have used these chemicals to study the changes of cells by diseases ranging from cancer to muscular dystrophy I mentioned earlier. And they use these chemicals to detect food spoilage and bacterial contamination also. And they use in spacecraft instrument because they can easily detect it at night using this bioluminescence. So these are the some other applications. And here, not only that, fireflies have ecological value. They are important as indicator species. Fireflies are good environmental indicator. So therefore, it is important to measure the quality of the environment. So it is a kind of good ecological value of the fireflies. And now I am moving to the next important aspect of the ecological value of fireflies. They are important as a poisonous insect. As you know, fireflies emit their light to find their mate as the ultimate aim, but they use these signals or the lights to warn their predators about the distastefulness and the unprofitability. So, and some scientists have found that the ingesting fireflies can be lethal to lizards and frogs. These are the some aspects of the fireflies that are important in environment. And there are some species, they are specific species, they use a kind of a defensive mechanism to avoid their predators, because these some species can release blood like uh, liquid. It is a bitter tasting, and also these chemicals are poisons to vertebrates, especially lizards and birds. So these phenomena we known as a reflex bleeding. Okay. So now I am moving to the last important aspect of the significance of fireflies. Their aesthetic value. The various types of firefly creatures have been developed now, especially the ornamentals, the visiting cards, nail decorations, toys, and puppets, and also these kind of creatures of the bioluminescence for firefly uh, fireflies is using the fireflies aesthetic value. And also there are so many firefly watchers and the interest people lovers, they pleasure to and they admire to firefly watching. So these are the some important aspects of fireflies. So here you can see some decorations using fireflies. I think I can remember my childhood when seeing these photographs like this. Some of you also have your memory. Okay, so these are the some creatures. Okay, now I am moving to the next part. Second phase of my talk, the status of fireflies in Sri Lanka. As you know, actually we are neighboring countries, and Sri Lanka also belong to the same hotspot on the with the Western Ghats. No, I think you may also happy to know about the Sri Lankan fireflies. First, I would like to talk a little bit on the past studies. When talking about the Sri Lankan fireflies, actually it goes back to the uh, 18th century in the colonial era. So many scientists and naturalists and other researchers came to Sri Lanka and collected specimens of animals including insects and fireflies during their voyage, and these specimens now have been stored as a repository firefly collection at the National Museum, Colombo, Sri Lanka. But problem is their museum collections very old and not yet updated. Not yet means to the to 2009 they not updated. So now I am moving to the next slide because uh, I want to highlight here some historical publications of the fireflies in Sri Lanka. The earliest record, the list of animals in Ceylon, published by Tennant 1861. It also included 28 species of fireflies in 5 genera. And thereafter, fauna of British India published. And in 1912 and 1973, two authors, Green and Bertrand, published 29 species of fireflies. Actually, their reports have included 29 species of fireflies. And as mentioned by the Dr. Ballantyne, McDermott list 1966 also reported 30 species of fireflies from Sri Lanka. The important aspect is among them 24 species have been originally recorded from Sri Lanka. That is the important aspect when searching the literature of fireflies in Sri Lanka. Okay, the major taxonomic impediments we face is the European naturalists have been originally described these specimens over 200 years ago, that means early 18th century. But thereafter, this fauna had not been explored to any extent up to 2009. Actually, after 2009, uh, we tried to re-record them, or so far we have identified 12 number of 12 species of fireflies clearly. Uh, these are the problems we face. Further, when trying to identify fireflies or systematic investigations of fireflies in Sri Lanka, I face these problems. Actually, these encountered problems are not reliable past taxonomic studies. That was a major problem I faced. And non-updated repositories at the museum collection. As I mentioned earlier, our museum collection is very old, it's not updated. And unknown type localities. As mentioned by the Dr. Ballantyne, our types are not here, they are in European museums. Considering the access and the we couldn't go there, actually it is somewhat difficult. Okay, these are some problems we face, and lack of reliable literature on the fauna in the region, it is another case. Okay, and you can see these are some photographs of the museum collection. Some specimens are not there, only labels, and there are some disorganized specimens here. So these are some problems we encountered during our study. And actually, I want to mention here the present study. That means since 2009, this is the team. We started the fireflies research in Sri Lanka. I really appreciate my supervisors. These are Professor Hemant and Professor Chitra, yeah, and also myself. We started the long-term survey on the taxonomy and ecology of fireflies in Sri Lanka 2009. We identified most important first step was we want to revise the classification of the repository firefly collection in our National Museum, Colombo, because as I mentioned earlier, their classification system is very old. So within this 200-year period, actually there were so many taxonomic revisions in the region and also the species. Therefore, we went there and take photographs and collect the specimens. However, we have not chance about to dissect them because they not give permission for that. However, we identified them morphologically and also classified their uh, classification system using the updated taxonomic knowledge in South Asia. And then, as a second step, we focus our study to the field to reassess the firefly collection or the firefly fauna in Sri Lanka by the collection of specimens from all 9 provinces of Sri Lanka. You know, Sri Lanka is divided into 9 provinces. We collected specimens from all provinces using 3 types of habitat: grasslands, paddy fields, and freshwater associated lands. You know, actually we selected these grassland terrestrial habitats and the freshwater associated habitat based on their literature, because you know fireflies have aquatic or the some kind of terrestrial habitat they live. And in addition, we selected paddy fields as a because it is a continuously disturbed by mankind activities, and during our preliminary study we observed that fireflies are common in these 3 habitats. That's why we select these 3. However, although initially we selected these 3 habitats, now we have widened up our research to the other habitats, the coastal habitats and some mangrove habitats also. I would like to talk a little bit on the materials and method, because sometimes it's important for the young researchers who are willing to do research on fireflies in your area. We selected these 9 provinces: was Sabaragamu, southern, western, central, eastern, northern, northwestern, and north central. I will show you the map in the next slide. And we selected 3 subsampling sites in each province: terrestrial grassland, paddy fields, and freshwater associated land. So this is the map of the Sri Lanka. It represents 9 provinces in different colors, and these are the locations we selected for our sampling. And this is some photographs of the selected habitats. And in this table you can get idea about how to document it, that these are the provinces and sampling localities and their GPS locations and their habitat. So actually when we are studying fireflies, I think studying of the vegetation type is also important, because insect plant interaction is a kind of emerging research trend in this region. So therefore we know actually there are a lack of information on these aspects also. Therefore, and also firefly larvae, always the behavior of the firefly larvae and their abundance sometimes depend on the adult's behavior. So therefore studying the finding of their proper habitat is important to find their immature states. And we collected samples from 5:30 to 10 p.m. in each sampling day. We selected 100 square meter area in each habitat and collect fireflies using standard size hand net. However, actually you know that in our population not much abundant, therefore we can easily collect them to the transparent polythene bags within this period. Then we identified as much in the field, uh, and females. Sometimes flightless females also we found. These females and larvae were collected using fine forceps when they were associated with males in the same habitat. That is important. And collected fireflies are temporarily put into the transparent polythene bags in the field, and they identified generally in the field using the morphological characters. But I am actually I want to tell you that if we are trying to identify, if you want to identify them in the field, even temporarily, you should enough familiar with your fauna. That means their dorsal and ventral color pattern, shape of the light organ, number of light segment. It is depend on their sexes. However, then you want to get some samples of these specimens to the and bring to the laboratory, then confirm. And a limited number of samples of both sexes and larval stage were preserved in 70% ethanol medium. For further identification and confirmation, we brought these specimens to the laboratory and then followed their identification features using the taxonomic keys and did the dissections also. However, according to the rules and regulations of the Department of Wildlife Sri Lanka, they considered as fireflies are rare animals. Therefore, they not given the permission to kill more specimens. Uh, this is the one kind of limitation. However, we collected males and females and larvae were taxonomically identified in the lab using available keys. In addition, you have to dissect them their light organ because it is important to confirm their taxonomy. Then again, I advise you, you can get the guidance or the supervision from other experts from the world to confirm your species. Okay, now I am moving to our findings. First important thing is be able to reclassify or the update the repository firefly specimens. Update means not the specimens, the classifications of the repository firefly specimens in the National Museum, Colombo. Actually, initially, our museum collection arranged into 4 families under 27 genera and 63 species. But we rearrange it 4 families containing 29 genera and 61. And we publish this information in this journal, Ceylon Journal of Science. You can find more information if you're interested. Then I am moving to the second achievement. Actually, from 2010 to present, we are able to identify these species, these specimens, actually 12 specimens. You can see here, we collected Abscondita perplexa and Abscondita pramalina under genus Abscondita. You know this genus introduced recently by Valentine 2013, and we identified Asymmetricata humeralis under genus Asymmetricata. This genus also introduced by Valentine recently, and Curtos costipenis. In here, I want to mention you that this species we recorded for the first time in Sri Lanka in our study. Before that, there were no any species recorded in Sri Lanka under genus Curtos. And we able to identify 3 Luciola species: Luciola extricans, Luciola horni, Luciola nicolari, and next one is Sclerotia satriata. Initially, this species be known as Luciola cingulata. However, Valentine, I think, am I correct, in 2009, introduced this genus as a combination. So, therefore, our Luciola cingulata species also belongs to this genus and species. And be able to identify 2 Diphenus species, Diphenus lutencens and Diphenus vitrifera, and Lampigera tenevosa. Okay, now I am going to show you a little bit about these fireflies in Sri Lanka. First one, Abscondita perplexa. Their habitat, we collected these specimens from 3 habitats, all habitats, and all over the country, especially throughout the year. It is very common in Sri Lanka. You can, however, when we are trying to identify them, you may face some difficulties because orange pronotum with elytral apices, but you can dissect them and get the further advice to confirm this species. And especially they have a kind of a incomplete dark band in abdominal ventrite of 5. And this is their larvae recorded associated with their habitats, and you can see their eggs also here. Then second one, Abscondita promelina, it is a kind of a pale firefly with black mesocuculum area, and when comes to the ventral side, you can see here their abdominal segment 5 is black. And however, I also found some specimens with both 4 and 5 segments are black, but belongs to same species. This species recorded from 3 provinces from Sri Lanka. And next one, Asymmetricata humoralis. It is also recorded throughout the country from terrestrial grasslands. However, you can see this, their body size is somewhat large, and their elytral apices and the base are dark. And when coming to the ventral side, you can see they have bipartite light organ in the abdominal segment 7, and their sternite 8 is asymmetric shape. These are some morphological features to distinguish them, but you have to dissect to confirm their species further. This one, Curtos costipenis, recorded for the first time in Sri Lanka, not previously. However, they recorded from terrestrial grasslands in Uva Sabaragamuwa and Uva southern and northwestern provinces. There are some specific characters on their elytra. You can identify when we are trying to identify them. The punctures and distinguishing intestinal lines and humoral carina. I think you can study further if you want to get more knowledge. That means these are some specific features to identify them. However, the ventral side is black with milky white light organ. And Luciola extricans. This species also original described from Sri Lanka. Interestingly, you can see here they are pronotum with central or the medium black patch, square shape, and black mesocuculum, and 1 to 3rd of the elytra are black. And there are intestinal lines here and somewhat deep punctures. This species recorded from 2 provinces so far, Sabaragamu and Southern Province. And it is a kind of other interesting firefly we record from Sri Lanka. It also originally recorded from Sri Lanka. From paddy cultivations in Uva province. Unfortunately, we couldn't find more specimens. That was the reason to missing of this abdominal part. It is because we surveyed their site continuously, but we couldn't find more specimens, unfortunately. The special thing is they have black elytra, black mesocuculum, and a dark black patch on the pronotum. And Luciola Nicolorini, another interesting one, we recorded very recently, I think 2 months ago, after their original description from the southern province in Sri Lanka. You can see here their elytra also black, however with orange margin, and pronotum mesocuculum also orange. Okay. However, ventral side you can see some kind of triangular pointed shape light organ there with black abdominal segments. Here you can see the sclerotia substriata. They recorded from freshwater associated habitat from 3 provinces in Sri Lanka so far. It is a kind of large, or they have large body size, similar to the Symmetricata humeralis. They also have fine punctures on the elytra. Especially, they can easily identify using these V-shaped light organ. V-shaped light organ. It is interesting. And Lampigera tenebrosa, it is also common in India. Uh, it is common in Sri Lanka. We recorded this species from terrestrial grassland habitats throughout the country. Uh, the larvae of this species, I mentioned earlier when I was talking about the environmental significances, especially the agricultural value. Their larvae we tested for the predatory value using snails and slugs. So these are the photographs of their female. You can see here. This is a mature female. They also have light organ, uh, and their larvae here. This is the larvae, and this is one of egg. And Diaphanous luteus, it is also recorded from Sri Lanka, and original recorded from Sri Lanka. This species we found from terrestrial grassland habitats from 3 provinces. Interestingly, they have a transparent areolate area on the pronotum here, uh, but their body is dorsal body is light brown, ventral black, and you can see here strip light organ here, 2 strip light organ. This is the female of the Diaphanous luteus, and the larvae we found associated with their original habitats. And the last one, Diaphanous vitrifera, it is also have transparent or the areolate area, and especially their body is transparent or the light brown, dorsal and ventral, and you can see here strip light organ also they have. We recorded this species from freshwater associated habitats in Uva province. These are the species we recorded or we identified clearly from Sri Lanka. This is their larvae. Now I am moving to the third achievement of our study so far. For the first time, we able to introduce the firefly fauna, the information of fireflies in Sri Lanka to the National Biodiversity Report of Sri Lanka for the first time. Actually, it is a sixth national report to the Convention on Biological Diversity. It is a kind of good achievement of our study. These are the some photographs of the pages inside this publication. Firefly beetles we able to introduce for the first time to this national and the international report. And let me introduce what are our future plans. Uh, we are planning to study the plant firefly interactions. Uh, that means associating vegetation and plant types of fireflies, and we are planning to study the behavior of fireflies, especially flashing patterns, differences among species, and we are trying to give a molecular based approach for identifying these species, and already we have identified some areas for the firefly based tourism or the conservation programs. We have identified some special habitats and microhabitats of some fireflies, so we are going to conserve them future. And also we are, as I mentioned earlier, we have developed some firefly based ecotourism areas for thousands of firefly watchers or the tourists. These are the things I want to discuss with you. Then let me show you our publications. These are the publications in index journals regarding the fireflies in Sri Lanka. And these are the conference papers. Okay, this is a summary. So far, I discussed the significance and the status of fireflies in Sri Lanka. This is a message for you. The significance of conserving fireflies is important. Okay, thank you very much for giving me a chance and for your audience. Okay, thank you very much.
Yeah, yeah. Thank you, Dr. Chandana, for your very enlightening work and also for telling us so many things about fireflies. And what we feel is that there are a lot of similarities in the firefly fauna between the two countries. And definitely we hope to be in active touch and collaboration. Now, there are certain questions which the participants are asking. May I invite Nidhi Rana? Can you ask any question? Yeah, Nidhi Rana, can you ask any question? Yeah, she is asking a question whether the fireflies, the light organs are present in the abdomen. In the presence of oxygen, they inhale, but how that reaction takes place in eggs. So she's interested to know how actually the eggs become bioluminescent.
Yes, I think it is also somewhat happened because you know the eggs have some spores. I don't know really. My suggestion is it's also have some minute spores and it can happen inside the eggs also because it can transfer the oxygen. No?
I see. Yeah, because it's also a living entity. Nishant Venugopal is asking, is there any citizen science efforts on fireflies around the world or in Sri Lanka? Citizen
Sorry, I
science.
can't.
Yeah, is there any popular club or association which takes care of the conservation and protection to firefly sites in
Yes,
Sri Lanka?
actually, not official. That means we are going to implement the project with the wildlife department of the wildlife, and also,
Yeah.
however, we couldn't get the grant here, but we are identifying some areas regarding the fireflies conservation. So far, then we are going to implement it.
Okay. Another interesting question is since India and Sri Lanka, there are very many forest dwellers, and we also have tribes. Is there any relationship between the fireflies and human medicine? Can the extracts of fireflies be used against deformities in human beings? That's another question.
Yes, I think because as I mentioned earlier, because especially the therapeutic purposes I mentioned earlier use fireflies as fish. You know the fish tank they also use for the admire for
Okay.
that means it is kind of therapeutic. Likewise,
Uh,
fireflies also used, but we have not used. There are some evidences, not in other areas of the country, but not in Sri Lanka.
I see. Yeah, one more question of Nishant is that what do adult fireflies feed on? What
No,
is their food?
scientists believe that actually most of the adult firefly do not feed at all because they believe that the gain energy from their larval period because it is a protein diet, no. So therefore, it is enough for their survival. However, in other countries, in European countries, especially Botanist species, they are prey, they are predators on their some species, especially on their same species. They are predatory fireflies, but common thing is they not feed uh, at all. So that is the thing.
Yeah. And another question of Nishant is that can we identify fireflies through photographs?
Yes, that means I mentioned their flashing patterns is species specific. That means it is species specific. So we can identify them using the photography, but not only photography. You should identify them using the taxonomy keys also.
Okay. Now, one of the questions from Sunil is that what's the general time period of larva in the life cycle of the firefly?
No, in my experience, I observed that they come out from the debris with the dusk. Actually, it is depend on the time in your country. I don't know. In our country, with 7 p.m. or to 2 p.m., it is an optimal time period. But after 2 p.m., we couldn't see them, especially
Oh,
larvae and the pupae and eggs.
I see. Yeah, another question, Dr. Chandra, is that under what circumstances do you think fireflies can serve as pollinators, especially in cultivated ecosystems?
Actually, I don't know about the pollination process because
Yeah.
there are some published papers in other European countries. Some fireflies feed on pollens. I don't know their role. However, I don't know the process about the pollination. That
Okay.
means the contribution of fireflies to that process.
I see. Yeah, another interesting question is that can we identify fireflies through their pattern of switch on, switch off, or through color of the bioluminescence they emit?
Yes, actually, I mentioned earlier the synchronizing fireflies not, I think so far not recorded in our area, but it is a kind of specific pattern we can identify, and also the color important because in our country I observed that some fireflies blink their light in orange, some yellow, some green, likewise.
Okay. Yeah. Uh, uh, Aishwarya is asking one question. How a layman can bring and grow more fireflies in his backyard? How a layman or a common man can bring and develop a small firefly park or a group in the backyard or is in his garden.
Yes, first you want to identify the existing fauna, then existing firefly fauna, then you have to identify their specific habitats, no? Specific habitats, then you can separate these areas for firefly watchers actually. Likewise, not disturbing their original habitat, it is important. Without artificial lights or without pollutants, you can manage it.
Yeah. Is Dr. Chandra, is any firefly park or a garden established in Sri Lanka? Have you been able to do that in Sri Lanka?
Yes, actually, we have initiated it in southern part nearby the our university. One of
Yeah.
wetland we have established, but actually in the past 2 years it is not implemented due to the lack of tourists. But there are some local tourists. We also going there for our research and to see fireflies. Uh, the southern part of the Sri Lanka in Matra, yes.
Okay. So thank you very much, Dr. Chandana, for your very delightful and interesting delivery. And we see that more and more questions are streaming, and it is for the information of all the participants that the speakers will take time to answer. And when they can spare some time, definitely they would love to answer to your questions. And once again, Dr. Chandana, we'll be in touch with you, and thank you so much for your time and effort.
Thank you very
Okay,
much,
yeah, we'll
Professor
see you
Ashwin
again.
Chakra.
Yeah. Ladies and gentlemen, we'll move on to the third lecture. That is by Dr. Devanshu Gupta. Now, Dr. Devanshu Gupta is in charge of the Coleoptera section of the Zoological Survey of India, Kolkata. Dr. Gupta's expertise mainly rests on the taxonomic studies of beetles in the Indian subcontinent, including Daman, Diu, Dadra, and Nagar Haveli Islands. He has over 12 years of experience in field collection, identification, preservation, and curation of museum specimens, especially beetles. Today, he has agreed to deliver a lecture on diversity of Coleoptera in India with an overview of the family Lampyridae, on which very little work is being initiated. Dr. Gupta also has facilitated our visit to ZSI, and he is very much helpful in collaborating with us in making progress the work on fireflies. Dr. Gupta, we handsomely thank you for your support and collaboration, and we would like to continue be in touch with you, and you have agreed to our invitation. Again, we are so grateful to you, and please, you may now deliver your lecture. Dr. Gupta, please.
Thank you, sir. Is my voice is audible, sir?
Yes, you are very clear.
Thank you very much, sir. Very good afternoon to all of you. Respected Dr. Chakravarti, sir. First of all, I would like to thank you for giving me this opportunity to present about the coleopterans of India to the in the Firefly webinar. Before starting my lecture, I would like to first thank my previous speaker, Dr. Leslie Valentine, for her excellent talk on Lampyrids of Southeast Asia. That is actually enlighten us and that will motivate us for future work in taxonomy of Lampyrids in India. And my colleague, Dr. Damika, for his Sri Lankan fauna. The topic of my today's talk is diversity of order Coleoptera in India, and an overview of family Lampyridae. My major focus will be on the diversity of Coleoptera in India. I am Devanshu Gupta, placed in Zoological Survey of India, Kolkata, and currently I also thank my current director, Dr. Diti Banerjee, madam, for deputing me for this webinar. So, as we all know that Zoological Survey of India is one of the organization in India which is responsible for calculating or estimating the faunal species to different government organizations and doing the taxonomic work since 1916. And we are cataloging the species at the level of states and biogeographic zones, and also in the protected areas like national parks, wildlife sanctuaries, and conservation reserves and the community reserves. Since till today, we have in India we have 1,03,258 species, faunal species from protozoa to mammalia known from our country. This I am just giving you the brief how diverse is our insects and how diverse are our beetles in our country. Subphylum Hexapoda alone includes the 65.4% of the overall fauna of our country. And these catalogs are wildly species known so far from India. Four subclasses are known from India: collembolans, proturans, diplurans, and the insects. Among insects, 26 orders are reported from India: Coleoptera, Lepidoptera, Hymenoptera, Diptera, Hemiptera, Orthoptera, Thysanoptera, and Rhodonata. They majorly constitute the 94% of the insects, like other parts of the world. 6% orders, 6% species belong to 18 small orders. I will be giving, this is the table. You can see the number of species known from our country in different insect orders. We have a total 6.2% of the global insects distribution in our country. And many of these insect orders are not being, means lack of taxonomy, expert taxonomists. Many of these insect orders are not being touched since long time. Like Embryoptera, very few works have been done in India. Like in Plecoptera work is going on, like Strepsitra, Megaloptera, Raphidioptera, all these groups, Siphonoptera, Mecoptera, all these groups, very few works have been done in, even in Coleoptera, many of the families, much work has not been done. In case of Coleoptera, you see it is the largest order not only in the world but in India in terms of diversity. 22,334 species of beetles are known. This database we are making in our section from different literature, published literature, and this always needs to be updated after some time. So we have 114 families of coleopterans known from India, classified under 19 superfamilies and 3 suborders. One suborder that is not represented in India, that is Archistomata, Myxophaga, Ediphaga, and Polyphaga. So out of 4 suborders known from 3 are known from India. Out of 176 families, 114 families are known from our country. So beetle fauna since timeline, I would say Carl Linnaeus described 50 species specific distribution from India. Then currently the total number of species are 22,334. Out of them, nearly 3,000 species are endemic to India, means they are not distributed outside the political boundary of our country. Out of this 22,000 species. Beetles, with contrast to endemic fauna of our country, beetles represent 10.9% species as endemics and 11.6% of the total endemic invertebrates. Before starting beetles is a huge order to study. There are, I am specialized in family Scarabidae. I am working in superfamily Scarabeoidea. My PhD was on dung beetles. Then these are some of the equipments, like we use mercury bulb for collection, then pitfall trapping, sweeping, and all these things we use for collection and preservation of the sweeping net. Then aquatic collections like Diticidi, Gyrinidi, Hydrophilidae, all those things we use aquatic nets for collection, then pitfall trapping, then beating the bushes. Wherever you go, you will find the beetles. You need to apply the proper methodology for the collection of the your group of interest. These I am just telling because many of the people might be starting their career in taxonomy or at the MSc level or the PhD level. So they should know proper curation, how the specimens are important, how they are being labeled, how they are being pinned. So you can see there are many labels. There are when you collect the specimens in the field, you properly label them with their state, with their habitat, with their latitude, longitude, date of collection. All those things you need to write down when you are doing some PhD or MSc dissertation. Then the permanent labels will be added after the proper preservation. Then photography, and this is the drawers, insect drawers. We keep the collections which are catalogued and in the family wise manner. Identified specimens are catalogued in the family wise manner. Then unidentified specimens we keep them separately till they are not identified. You need to have different sort of vials also for collecting these beetles. If you want to preserve them in cool or dry preserve them, then microscopes you will be requiring for identification or illustration for making the publication in good taxonomic journals. This is very important for us taxonomists. We need to have a good photography for making the publications in good journals like Jutexa or Jukis or Coleoptera Bulletin, etc. So the major work which we are still I will say that we are identifying fauna of India in case of beetles from the fauna of British India volumes which were published date back in 100 years back. Cyrambicidae we are identifying by Gahn, then Chrysomelidae, then Scarabidae by Aero, then Cicindalidae. The tiger beetles we have good field guides also, then Marshall, Corconudae, except these are the preliminary publication and the basis of the publication, but many of the revisionary works and monographic works after those have also been published on this group. And the Coleoptera catalogus, as Madam Dr. Valentine told that we need to have a catalog of all the, before starting any group, we need to have a catalog of all the species. That's why we need to have this Coleoptera catalogus, not only the old series as well as the many of the word catalogs have also been published in case of beetles also. So this is the diversity, the insect, the beetle diversity in India. The Orchoestimata is still not reported in our country. Out of Myxophaga, 5 out of 4 families, 2 families are reported from our country, and out of 104 species, 7 species are reported from India. Edifaga is 12 families in the world, 7 families are reported from India, and 45,841 species are there in Edifaga, and out of them 3,045 species are known from India. Among Edifaga, Carabidae is the most diverse family. Polyphaga is of course the most diverse insect suborder. 105 families are known in this suborder, and over 19,000 species in this particular suborder are known from our country. Major coleoptera families, I would say that this Staphylinidae, that rope beetles, Chrysomelidae, the leaf beetles, Carabidae, the ground beetles, Curculinidae, the weevils, Scarabids, the dung beetles and chaffers, Cerambicidae, the longhorn beetles, Elateridae, the click beetles, and the darkling beetles, Tenebrionidae. These 7 families are the most diverse and constitute more than 12,000 species in India. Now, coming to the beetle diversity in different ecosystems in our country, we have published this information in the in different books in the form of Fauna of Ecosystem series. From you would like here to mention that from agroecosystems, 359 species of beetles are known. These basically 359 species are basically pests and predators of in the agroecosystem or agriculture crops. In the freshwater, we have 776 species known from our country. Out of 22,000 species, 776 species in 17 families. 17 families are associated with the freshwater ecosystem. And in soil ecosystem, 8,862 species are have been documented from India. I would like to here mention that 340 species exclusively are associated with the mangrove plants, which are in the Sundarban areas, in the western coasts and the eastern coasts. This Himalayan region, because the most of the discoveries and all those areas have been done from the western and northwestern Himalaya, we listed that 10,533 species of beetles are known from our Himalayan region. Half of the diversity is known from the 6 states from the country. That is Uttarakhand, Himachal Pradesh, Sikkim, Darjeeling area, and Arunachal Pradesh, including the Jammu Kashmir and the Ladakh. Cold desert, that is the Trans-Himalayan region, we have only 93 species of coleopterans known from, so far known from cold desert, Trans-Himalayan region. From desert, that is the Rajasthan part, 12 districts of Rajasthan, the western Rajasthan, only 326 species are known. And from the island here is the this Andaman and Nicobar island. We have 936 species of coleopterans known from this particular area. You see the island is Andaman Nicobar island is having only 2.25% of the area, but includes so much diversity. This is quite highly diverse area in terms of coleopterans in our country. I have also because it's not much time is not much. This slide itself is will be giving the overall information regarding fauna of different ecosystems and all those area. You see here in the Himalayan region, overall fauna known from the Himalayan region is 30,615 species, and out of them 10,000 are beetles. Then 1/3 of the diversity in that particular ecosystem is of only beetles. But how many taxonomists in India are working in beetles? That is very means that is questionable. Very few people in India are working in taxonomy of beetles. Most of the works have been done. So as this particular slide, as India is divided into 10 biogeographic zone. This is the classification of India is divided into 10 biogeographic zone. The upper one is the Trans Himalaya, then the Himalaya, Northwest, West, Central, and the Eastern Himalaya. This portion is the Himalaya. Then the Northeast region, then the Gangetic plains of Uttar Pradesh, Uttarakhand, and the West Bengal, then the peninsular India. And this part is the semi-arid India, and this one is the our Rajasthan desert area. Then the Western Ghats and the Andaman Nicobar. While making the inventory of beetles in different of India, we also gave their distribution in the biogeographic zone label. Then we came to the conclusion that that most of the beetles are distributed in this our Himalayan region, and then our Himalayan region. 47.2% of the overall diversity of India is reported from the Himalayas. And when we compare with the total area of that particular region, 0.25% island is including 4.2% of the beetle. And in case of you see Deccan Peninsula, the peninsular part, this portion, this includes major part of the country, that is 42% of land area of our country, but the diversity you see is only 14%. And this also includes the overlapping species with the Gangetic plains and the semi-arid area. Western Ghats include 1,977 species. We also, while giving the checklist, we also distributed them different states. That is a political boundary. India is divided into 35 states and union territories. Now coming to the suborder level diversity in India, the suborder Myxophaga, this is the first order. Archaeostomata is not reported from India. Two families are reported in this particular suborder. These are called skip beetles and minute bog beetles, very minute in size. Only 7 families are reported: Hydroscaphidae and Sphaericidae. These thrive on the algal mats, and these are aquatic beetles. These come under water beetle category. They form thin film, particularly on the edges of running water. We do not have classified specimens of this particular suborder in our collection. This I took from the suborder Ediphaga. This is one of the important group in our ecosystem, that these are basically carnivores and omnivores. There are 6 families in this particular suborder reported from India. That is Gyninidae, walling beetles, Rhizodidae, Carabidae, Heliplidae, Notaridae, and Diticidae. We have a very good collection of Gyrinidae, and this Gyrinidae we have also published fauna of India, which can through which anybody can identify all the gyrinids from India. Carabidae is a big order. One of the person in my section is also working on this particular group. We have a huge collection of Carabidae and Heliplidae, Notaridae, and Diticidae. These are we have more than 200 species in our collection only. 250 of Diticidae are known, and we have 200 in identified species in our collection. These Carabidae play a role of carnivore in the soil ecosystem, and these aquatic beetles in this suborder are scavenger or detritivore in ecosystem. Coming to this is suborder Polyphaga. Suborder Polyphaga is the largest. I already told more than 19,000 species are known from our country. Suborder Polyphaga is classified into different superfamilies. Those superfamilies we are superfamily Hydrophiloidea. In this these are detritivores in aquatic ecosystem and also as a predator. Hydrophilidae is the largest family in the superfamily Hydrophiloidea. Then Helophoridae, Epimetopidae, Spergidae, Georicidae, and Syntelidae. These are the least diverse species, diverse families in India. Then Hysteroidea, there are more than 260 species of hysterids, I would say, are known from our country. These are highly predaceous beetles feeding on maggots in dung, decomposing and organic. Here I would like to say that hydrophilids, some of the hydrophilids are also associated with the dung also. They are collected with the dung pads also. Now, this superfamily Staphylonoidea, this is the largest or most diverse group of beetles in India. There are 6 families in this group also: Hydranidae, Telidae, Egeretidae, Leodidae, Sylphidae, and Staphylidae. Staphylidae, very big family. This rope beetles is a big family, and I don't think anybody in India now is working on this particular family. One previously in my section, Avasar was one of the scientists, Mrs. Avasar was working, but now nobody in India is focusing on this huge family. We should need so much trained coleopterists in India for working in this group. And the Egeretidae and Sylphidae, these are forensically important groups. They mainly feed on carrion. Hydranids and leodids are commonly known as minute moss beetles and the fungus beetles. Staphylidae are mainly ground dwelling beetles. Now coming to the superfamily Scarabaeidea, they are scarabaeids feed on dung pads. Basically, there may be 2 categories can categorize. Carrion feeders are also there: dung beetles and the chaffers. So dung beetles, there are 7 families known from of Scarabaeidea. This is the group I study. My area of taxonomy expertise in this particular group: the Geotropidae, Phasalidae, Trogidae, Leucanidae, Hypoceridae, Cocodidae, and the Scarabaeidae. They help in nutrient cycling, parasitic suppression, secondary seed dispersals, and the plant growth. This is we recently described this geotropid Bulboceras arunachalensis from Arunachal Pradesh, and the Puku arunachalensis from Namdapha National Park. This is a Rutalini, and this is a geotropid. These are leucanids. We have very good identified collection of this particular family in our section. Trogids are very less diverse. There are only 17 families species known from India. Likewise, Hybosorids also, and the Phasalidae, 28 species of this group is known from our country. The superfamily Scartoidea, this consists of families Scartidae and Chlamydidae. They are found in detritus and fungus-covered tar barks. The terrestrial beetles are found in vegetation close to water bodies and water-filled areas. So there are 98 species of this superfamily. Scartidae includes 80 species and Chlamydidae 18 species. Now the superfamily Dactyloidea, only 1 family in this particular superfamily is reported. They are found in moist soil and under rocks. Adults are often found in grasses. 17 species of this particular family Dactylidae is known from India. Buprestoidea, these are jewel beetles. Larvae burrow through roots and logs and stem, and they cause huge economic loss also to the dead trees. 604 families of buprestid beetles. You say jewel beetles are known from India, but very few species have been identified in the published literature. In case of Indian taxonomy, in case of buprestid, very few 10-12 species are being identified in general phonistics. The other species are not being reported and identified because of the lack of coleopterists or trained taxonomists. Then the superfamily Biroidea. This includes families like Viridae, Elmidae, Dryopidae, Limnicidae, Heterosidae, Cephanidae, Telodactylidae, Chelinonaridae, Eulichidae, and Carididae. These are found in thermal pools, hot springs, subterranean habitats, and decaying woods. Among them, these Viridae, 30 species are known, Elmidae 28 species, and Limnicidae includes 62 species. The most This Eulichadidae and the Chilonaridae includes only 4 species, and the Telodactylidae 3 species. Now coming to the Elateridoidea, this includes 6 families from India. The click beetles and the fireflies, which is the topic of today's webinar, also belong to this this particular superfamily. But the fireflies, we have heard so many. We have been told about fireflies by the experts in previous 2 talks, so I will not much focus. We have cataloged 50 species of fireflies from India and their distribution. Elateridae, we have in this superfamily, Elateridae is the dominant family with 861 species. So fireflies, as Madam Dr. Valentine has explained much about these group of animals, and Dr. Dhamika also has explained many of the ecology and all those things, and the Madam has explained about the taxonomy also. So for the checklist, we have in our hand, we have 50 species in 4 subfamilies: Cyphocerinae, Siphonocerinae, Lampirini, Luciolini, and Otoretini from India. These are the genera known from India. The Lampirid genera known from India are this, and many of the species are in the original publication are listed only with India without precise location. And Madam and Chakoti sir, we are having a huge unidentified collection from Himalayan region, Northeast region, as well as all over India. We have a very good collection, but that is not yet being identified. When Mr. Parvez came to Coleoptera section, visited us, we started segregating the species into different morpho species. I asked them to classify them into morpho species, and then we can identify them through literature. Now, I would request Madam Dr. Leslie Barrington to help us in this identification of so that correctly the species can be identified. Now, this whatever we have the information, 2 species are known from islands, that is Andaman and Nicobar Island. Then 10 species are known from the Northeast region. 6 species from the Himalayan region, and the 2 species from the Gangetic Plains, and the 7 from the peninsular India. Only 1 species from the semi-arid island, that is from Delhi and the Western Ghats. 6 species are known. From the Rajasthan desert area, there is no record of lampirids in our database. These are the taxonomic significance characters which were used in lampirids that I will skip because much have been told by Dr. Leslie Barrington. These ecological significance much have been discussed by Dr. Dhamika. So it will be better to skip this thing. Firefly conservation. Actually, in India, many of the people ask that how can we conserve these fireflies. When Western Ghat reports and all those things reports, if we see that many of these firefly incidents are coming from the national parks, wildlife sanctuaries, and the conservation areas. We have 907 national parks and protected areas in our country. Through those areas, we can contribute towards the conservation of fireflies by the forest officers, DFOs, and these people. We can engage them, and we can do something for firefly. This is light pollution, huge pesticide use, climate change, habitat loss. These are the major things which are very important in the leading to the lower population of fireflies in basically our urban areas. We don't see much fireflies which we used to see in our childhood. Some of the beetles' most effective strategies for conserving, identifying, protecting, and restoring high quality habitat. High quality habitat is in India, that is the most populous country we have in the protected areas only. That is our 23 to 24% area is protected under Wildlife Protection Act. That we can be used for the protection of or the firefly conservation. Superfamily, I will come to superfamily Derodontoidea. It is include only 8 species. Nosodendridae and Jacobsinidae are the 2 families known from India. The species mainly occupy this underbark in the plant litter and rotten wood. Bosticidae, very important group of beetles. They are wood-boring beetles, mammalian conifers, and Tinides are scavengers, and dermestids are found in skin and dead insects. Dermestids, we have 99 species reported from our country, and the bosticids we have 63 species. We have made the checklist of bosticidae of India recently. The number of species known from India are 78, which is not been, I have communicated to the journal, which is under review. After these are the 78 species. Tinidae, 29 species are known from this, and bosticids are very important in timber and all those. They cause huge loss to our plants. Limaxilloidea, only 6 species are known from our country. These are basically ship timber beetles which invade forest plants as well as wooden furniture. Claroidea, only these 4 families. These are Trogositidae, includes 11 species, Claridae 175 species, Prinocertidae 7 species, Meliridae 141 species. This is the Cucuzzoidea, very important group. There are two scholars or staff working in this in my section in this group. This includes 1,294 species. In this, the ladybird beetles and the Nitidulidae sap beetles are very important group. All the we have the A.P. Kapoor's collection, Dr. A.P. Kapoor collection in our museum. 500 more than 430 species of ladybird beetles are known from India. And Nitidulidae, we have PhD in one of the scientists, Dr. Jikmik, has worked on that group and described many new species from northeastern India. And Dr. Priyanka Das worked on the ladybird beetles. We have and we have imaged and digitized all the specimens in our museum also. Now the Tenebrionoidea, they are predominantly detritivorous, herbivores, and Mycetophagous. Is a family. Tenebrionidae is the 5th largest beetle family after Curcunidae in India. These Meloidae, the blister beetles, also come under this group. Then Anthicidae, then Odimeridae, Tenebrionidae, all those groups come under this superfamily. Chrysomeloidea, this includes 4 major families: the leaf beetles, that is Chrysomelidae, then Cerambicidae, the longhorn beetles, then the Megalopodidae, and the distal. Cerambicidae, longhorn beetles, much can be identified, and with the many good websites are also available for identification. Field guides as well as the fauna of India is also. Chrysomelidae identification is we have fauna of British India published by Jacobi and Momik. That group is the largest among this Chrysomeloidea. Largest among Chrysomeloidea and the 2nd largest family in India after Staphylinidae. Curculinidae also very important group. These are commonly called as beavers. They are agriculture pest as well as forest pest of stored grains. Anthribidae includes 261 species. Atalabidae 242 species. Brantidae 353 species. Draphthoridae 35 species. Brachyceridae 4 species, and the Curculinidae 2,500 species. What we have in our collection in my museum, what we have, I have nearly 314,425 specimens of beetles, exclusively of beetles from all over India, from different biogeographic zones, from different states, all the, and among them 4,835 identified species we have with in our collection. Number of families, what we have in our collection is 76 families we have in our collection, though 114 families are reported from India in the literature, literature and the literature, but we have only 76 families which we have identified. These are some of the publications we have made in recent times on the family ladybird beetles and the Scarabeoidea, the geotropids, and all those. We have collaboration with Dr. Doug Ahrens also regarding the Sericini beetles. One of the scholar is working in the taxonomy of Sericini in our group, and she has described more than 9 species new to science. These are some of the compilations we made for freshwater beetles in India, the Indian Himalayas, then the state soil ecosystem, then the mangrove ecosystem, then the island, then the one monograph on the family Cupujoid and Tenebrionid families were published by the Dr. Tarun K. Pal. T.K. Pal is one of our senior colleague working on the Cupujoidea family. What we don't have, that is the gap area what we need to have in India. That is, we need to have a checklist of Coleoptera of India. That we don't have published information. Many of the things are being published by the global authorities in the group, and the taxonomic studies specifically at the genus level and the family level. We need to develop the this taxonomy. Why I am emphasizing? Because in India, taxonomy especially is not being much promoted by the administrative part for in the jobs and all those things. And we need to have the fauna at different ecosystem level. Then we need to have the long-term monitoring, like we need to have long-term monitoring of fireflies and the impact of climate change, like Dr. Chakravarti sir is doing in the Western Ghats. We need to do the geospatial modeling and the conservation biology of these species. For that, for achieving all those things, we need to make different institutions collaborate each other, share the specimen, share the information with each other. So then only we will be able to promote the taxonomy and the science in our country, and repeated sampling in the historical prominent collection localities to study the impact of climate change. That also need to be done, and the suitability modeling of the species need to be done. Then DNA barcoding has to be done by our part. This is our team under the leadership of our director, Dr. Diti Banerjee Madam. Myself is Devanshu Gupta is working in scarabid beetles. Then Ravi Kushwaha is working in carabids. Dr. Jikmik Das Gupta is working in Nitidulidae, family Nitidulidae. Mr. Asim Ghosh and Ms. Fulmani Mandal, they are responsible for the proper preservation and all those things, activities, labeling, preparation. And these are the 5 students working in different families. Dr. Joyjit Ghosh is working in scarabids. Dr. Priyanka Das is working in coccinellids. Devika Bunia, she is working in tribe Sericini. Shipra Sonali, she is working in aquatic beetles. And Priyanka Ghosh, Shipra Sonali and Priyanka Ghosh, they have recently started on aquatic beetles and the chrysomelids. Thank you very much for listening me. Thank you very much.
Yeah. It has been a very good journey for us and a very comprehensive overview of the coleoptera fauna of India, Dr. Devanshu Gupta. And definitely, as you have emphasized, we need to cooperate, collaborate, and exchange information. So that's very important, and that's how we can take the science of fireflies way forward. This is very much need of the hour. So you have rightly emphasized the areas which need to be strengthened in India, and everyone has to put in his share in order to do something significant as far as fireflies and other Coleopters fauna of the subcontinent is concerned. Now we have a couple of questions, Dr. Devansh Gupta, and hope you will be happy to answer them. I would like to know whether there is why the Zoological Survey of India has not formed a team to explore fireflies, especially in some of the high hotspots, high density hotspots of the subcontinent, like the Himalayas, the Western Ghats.
In that case, I have said that we have very few people working in Coleoptera. It's a big order. Like we are specialized, like sir, you are working in Lampyrids. Then for me, doing the Lampyridae identification and all those things will not be much much useful. Like you are doing and Madam is doing. Then we need to learn. And in case of JDSI has the collection, we are surveying entire Himalayan fauna, but it is not identified. We are doing the survey in the Himalayan region as well as in the Western Ghats. In the Kerala, there is one center that is in Kozhikode. Kerala, Kozhikode. There is one center. They are doing that work in the collection of different group of animals, including the fireflies also.
So how many species of beetles do you think are endemic to India, Dr. Gupta?
More than 3,000 species are endemic to India. More than 3,000.
Okay.
3,133 we have endemic to India, which we catalog in our database.
I see. Dr. Gupta, see, we have been experiencing a drastic decline in populations of Elateroidea beetles. And we have now very few what you call active populations of jewel beetles, the click beetles, yes, and including the Lampyrids in Western Ghats. So that's what we have been witnessing. There have been unabated urbanization and commercialization of these spaces. Now, do you think in Northeast India also such trends are noticeable?
Northeast India, these trade things are not much there, but there are these larvae and even the adults in some cases they are used as food. Northeast India, there are more than 100 species of beetles are being eaten by these people. But trade where trade is there, but marked trade are not there, because in Wildlife Protection Act we have only 37 species. Sir, you might only protected by the government. That is only 37 species, and those are very small ones. That Cucujidae, how that cannot be that is a pest. Some of the chrysomelids, these are pests. These have been protected. So through government, we need to have a proper information. No such information is there as in case of trade, we can say.
Yeah. Now one youngster is asking, is there any updated keys to identify Indian beetles or beetles in India?
Beetles in India, as you know, my fellow person is want to identify entire beetles. That will be very difficult task. Even to family level identification, good keys are available. Crowson's keys available. That is best. We need to identify. And but there is one book on T.K. Sen Gupta, and book is there, Beetles of India. There all the families can be identified. Then we need to have for species level identification, we need to have some training for the identification, taxonomic training, and we need to, if we are not expert, we always need to take help from the experts in the particular group. And now the things are global authorities are there to help us. We can also help in JDSI. They can come and compare the material. with the identified material. But the keys I cannot say. Complete detailed fauna 22,334 species key. It's a Herculean task to be done.
Yeah, sure. One of the group is asking, can you throw more light on Lagrine subfamily of beetles? Lagrine.
Lagrine in Elaterioideae.
Yes. Yeah. And throw some more light, he's asking. Yeah.
Much I have not much information in this particular
information. Okay. That's fine. And one of the things what the ZSI can do as we go about analyzing the taxonomy of beetles is that we can declare some of the good spots of beetles as hot spots. And I think ZSI can play a major role in this initiative. What is your comment?
Hotspots for beetles.
Yeah, beetles, especially. See, we know that in South India, no, in Annamalai hills and in Charmadiga Ghat sections, there are large millions and thousands of fireflies emerging. So definitely, this is a high time that at least to start with, we preserve such habitats.
Yes, we can do for species specific things like fireflies. And some of the species specific things we can do, like butterflies, something. Hotspots, there are already hotspots are there, and the classified hotspots are there. But species specific things we can see how can we do in case of beetles. Species specific, like in case of butterflies, we can do in case of that. And the will be helping means for the things, the scientific assistant, whatever we can do, we will be always be there.
Yeah, one of the student is asking that can you give some information about the keys to identify click beetles? Click beetles.
Elateridae. There is one website is there. I don't remember, but good Dr. Giovanni Plesia has published good number of papers. But the student has to concentrate on genus level, subfamily level. Keys are available at the generic level, species, genus level, then the subfamily level. The student has to get the key, overall key for the entire 834 species of Elateridae is not available. Not available. The genus level keys are available, subfamily level keys are available, but entire 834 species keys are not yet published. Huge work is to be done.
Yes. Now, one of the enthusiasts is asking, can we relate the identification of beetles to the plant species? So suppose we find beetles consistently on some plants.
Yes.
Okay. Now, can we take this as a cue or indicator for the possible identification of beetles?
Associated monophagous beetles are very less in nature. By seeing the plant, you cannot identify. Most of the beetles are polyphagous only. Then most of the beetles are polyphagous. Then identifying the plant and giving the name of the beetle, that will not be much helpful. But interactions are there. The beetles is associated with some plant or forest plantation or agriculture. That information is available with us.
So Dr. Gupta, we handsomely thank you for your time and sharing the so vast information in a secondly brief time, and we wish to be in continuous touch with you as far as the Lamparis are concerned, and we look forward to having more fruitful collaborative programs on fireflies in India. Then participants, all the participants join us to thank you very much, and hope we will be in touch in the times to come. Thank you so much, Dr. Devanshu Gupta. Now, participants, ladies and gentlemen, it's time we have our lunch. So we had very illuminating talks by Dr. Leslie Ballantyne, then Dr. Chandana, and then Dr. Devamshu Gupta. So we'll have a break and we'll resume this webinar at 2:45. That's just after 40 minutes Indian Standard Time. So hope to see you again. So enjoy your lunch. Thank you. Back again. What is that? No. No. Good afternoon, everybody. Hope you had a sumptuous lunch, and we are awaiting for yet another very interesting lecture on bioluminescence of fireflies, just in 2 minutes. Oh, Locke has joined from Vietnam. Go ahead, Barwa. So, ladies and gentlemen, good afternoon to each one of you. We will now begin the afternoon session. And we have with us Dr. Anurup Gohain Barua. Dr. Anurup Gohain Barua did his PhD from the Guwahati University, Assam. And at present, he is a professor of the Department of Physics, Guwahati University. Professor Barua's studies include beams through gaseous media, simulation of molecular spectra, spectroscopic characterizations on astronomical objects like the comet and meteorites, like the objects of bricks and fruits and trees. Professor Gupta's present interests are firefly bioluminescence and thermoluminescence dosimetry. He has published more than 50 research papers in peer-reviewed journals on this topic. He has guided 3 PhD students on firefly bioluminescence. Professor Gohain Barua is an Indian National Science Academy visiting fellow at the Rajaramana Center for Advanced Technology, Indore, and he is an invitation fellow at Chiba University, Japan, for the Japan Society for Promotion of Science. Professor Barua, we have been very lucky to have you with us because it's a science which few people venture, because we are just beginning to learn more about the biological significance of luminescence in living beings like the fireflies. So, Dr. Barua, please.
Thank you very much for such a nice introduction. And thank you for giving me this opportunity to speak before you. I assume that none of the participants are from the physics background, but I am a man of physics.
Yeah.
But okay, you need not be worried.
Yeah.
Too much of physics, I am not going to speak. But well, what I have been doing is that because I am a physicist, I am interested in the light and the flashes.
Okay.
And based on the emitted light and the flashes, we have been trying to make some conclusions about the light emitting reaction
Yeah.
and the light emitting system inside the body of a live firefly. Otherwise, what these chemistry people will do that I mean, you will have to prepare the analogs and do some chemical studies, or I mean, you will have to kill the insect
Yeah.
to
Yes.
bring out that light emitting part and carry out some chemical studies there. But that way, the real situation will not be there. So my objective has been to get some information about this light producing reaction as well as the light emitting system inside the body of a live firefly. So I have taken the help of this emitted light and the pulses. Pulses means that it's of course in different conditions, like say in normal condition at different temperatures, under a magnetic field, in an anesthetized condition, and so on. So based on this, so okay. So let us now go back to these slides. So is this visible?
Now present the slides.
Okay, so
And Professor Barua, you are not visible. So please make yourself visible.
so I have already removed this. Okay, I think okay, I'm not very expert here. Okay, I think my face is not that important.
Yeah, but yeah, still.
Okay, I will have to do something else then.
Yeah. Yes. Yeah, he will take time.
I do not know what has happened. I think we should continue with this if the slides are visible. Okay. So will this be fine?
Sure. Go ahead.
Excuse, sir.
Yes.
Can we present from ourselves?
From?
Because we have already one copy of your site, so that we can share if you want.
No, I think it will be enough. I think. I mean, if it is visible to you, I mean, if
Yeah,
it is
yeah.
okay.
Yeah.
Okay, so let us.
Please start the presentation.
Okay, so the title I have given is that it is information carried by the light of the firefly. So this is a philosophical statement. The firefly gives light to its pursuer. I hope so, as I have been running after it for last decade and a half. Okay. So these are among the most charismatic of all insects. And so many poems and songs have been given birth by this flashing of the fireflies, as well as the scientists also. So this is one poem from the early part of the 20th century. The firefly's flame is something for which science has no name. But well, at that time,
Professor Barua, 1 minute. Just sorry for
yes.
interruption. We are not able to see your slides. We are not able to see your presentation, please.
Okay.
Yeah, it's
so
not
means
visible.
that it is not visible.
Yeah, it's not visible, please.
Your page is visible.
Yeah, your page is visible, but your slides are not visible.
What has happened?
Yeah, just you can see if you can be very visible about your presentation.
So, so it is not visible now?
No, it's not visible. So, shall we start sharing? If you permit.
Okay.
Yeah. Just wait a minute, ladies and gentlemen. We'll just start sharing the presentation slides of Professor Barua.
And sorry for this problem.
No issue at all.
What is happening?
Yes. Yes, now we have started presenting your slides. Is it visible to you? The first slide, information carried by the light of the firefly.
I have not seen this much.
Okay,
Something is wrong somewhere.
I see. But we have just presented your first slide.
So, okay, I want to log out and then want to get in again. So,
Okay. I think he will be very, it will be convenient for him if he can make a presentation as well
what has
as
happened?
deliver the talk.
I don't know what has happened. It's been
Yeah. Just see, you try to resolve the issue. Meantime, we have kept your presentation ready. So that the participants can have a look at your slides.
I don't know what is going on here because it is causing some problem.
Okay. Then what we will do, Professor Barua, you go on with your lecture. We will be presenting your.
No, sorry. So it will not.
Okay.
I mean, what is happening here?
Yeah.
No.
Just you can. I think you can just log in.
Okay, we will start it again, all over.
Yeah.
So get started.
मेरा ऑडियो बंद कर दूं.
Screen sharing is paused.
Yeah.
I do not know why this message is coming here.
Acha, sharing is paused, is it?
Okay, I will stop share and then I will start it again.
Okay.
What is the problem here? Do you see this now?
No, we are not able to see. Screen is visible, but the presentations are not.
What is happening? You are sharing. I do not know what is the problem here.
Okay.
Rejoin.
Okay.
Rejoin. You can rejoin, Barwa. You can just rejoin. See.
Okay, I will.
Yeah.
Okay, I will do everything. Now.
Yeah.
Do you see me now?
Yeah. Yes. You are able to see. Yes.
Okay.
Yes.
So now.
Yeah.
I will try to do this screen sharing.
Yeah.
It is not coming. So do you see this now?
Yes, I can see. Yeah, we can see you. You are visible.
And the slide?
No, we are not able to see your slides. You are visible and you are quite audible, but screen is not being shared. Yeah, now visible.
Your page is visible.
Yeah,
okay.
Page is visible. Yeah.
Now?
Yeah,
Okay,
you can start. Yeah.
So is this coming?
No, your slides are not visible here again. Some issue may be there.
Okay, so is the second slide coming now?
Second slide. No, we are not able to.
Okay, then slideshow mode I will skip.
Yeah, you can speak. Yeah.
Okay,
So,
I
yes.
can speak.
Yeah.
Without going to the slides.
Yes,
Okay.
yes, please.
Okay, so is this now visible?
Yes.
Okay. So the Firefly gives light to its pursuer. So this
Yeah.
is a philosophical statement,
Yeah.
and I hope so. Okay, so many poems and songs have been generated from this, and this is one such poem. But of course, for which science has no name, but then definitely science has some name now. I can think of nothing eerier than flying around with an identified glow on person's posterior. Okay, so this is a very old poem based on this Firefly light. But see, so I am going to speak on about the emitted light and flashes and what information that light carries.
Yeah.
So bioluminescence, as you all know, that the living organisms convert chemical energy into light. So there are so many organisms that emit light: bacteria, fungi, crustaceans, mollusks, fishes, and insects. So many. So as you have already been informed today, the first half itself, it is the Lampyridae. Of course, I am not a man from zoology, but still this much of information I have somehow gathered. And about this number, 2,000 species, but today in the morning, I got to know that it is more than 4,000, I think. Our director today at inauguration, so he informed that it is more than 4,000 species. So, okay, so
Yeah.
this information has to be updated, I think.
Yeah.
Okay, about this reaction, so luciferin is the molecule, and the catalyst is an enzyme which is luciferase. I think this bit also you will know. Luciferin is the molecule, and luciferase is the catalyst. It is called chemiluminescence reaction. Of course, chemists are more interested, or rather, biochemists are more interested here. So it's a multi-step process. But well, because it is in the slideshow mode, it is not coming. So I have to show it as a whole. So our oxygen is the biochemical trigger. So that the substrate molecule is excited. Well, the luciferase will be needed. It is the catalyst. And well, adenosine triphosphate and the magnesium will be needed. So an unstable intermediate is produced, dioxetanone, and this intermediate, it decomposes. Then it is the one that we want, oxyluciferin. So this oxyluciferin is the upper step while decaying. So we have the emitted light. So I repeat that oxygen is the trigger. So luciferin is excited, and finally we get oxyluciferin. Of course, in between you have an intermediate. So luciferin is the molecule, luciferase is the catalyst. So of course, about the reaction part, the detail that, see, I am not a chemist also. So in presence of ATP and magnesium ion, so you have this intermediate is produced. Then it forms this oxyluciferins by splitting of this peroxide bond, you can call. So while decaying, it forms this oxyluciferin is back again ground state plus this light. So it is, in brief, this is the mechanism of this reaction. Of course, it belongs to chemistry part, and I am also not a chemist. I am a physicist only. Okay, so why it is so attractive for us? It's a very efficient system, meaning its loss is quite negligible. I mean, the minimum efficiency has been put at 41%. Of course, initially it is thought to be about 90%, but the minimum efficiency, meaning that its loss is still a very small one. So most of other light producing chemical reactions are only about 1%. That is 99% is lost as heat. But this is a cold system, so not much is lost. So that is where our interest lies. Because it is a very, very efficient system. There's hardly any loss. I mean, very small loss is there. So we can comment that the Mother Nature is clearly an efficient user of energy. Okay, so as far as I am concerned, so it is the emitted light and flashes from 4 Indian species of fireflies our group has studied, and 3 of those are identified by Dr. Valentine. So we are very much thankful to her for identifying these 3, and this is diaphanous. So here, Dr. Fu, but of course he identified only the genus part, and in his own word, the species is a problem which none of his group wanted to take. That was his joke. So it has to be diaphanous. And there are 2 Japanese species, Luciola cruciata and Luciola latata. Of course, these 2 were studied at Shiba University, Japan. So this is the one which is widely available in the northeastern part. I think it should be in the rest of India also, but in northeastern part of India, especially in Assam, in our campus itself, it is widely available. I mean, after darkness, you will see lots of firefly flashing. And this is the male, of course. It has 2 segments in its lantern, and the female has only 1 light emitting segment in its abdomen, in her abdomen. Oh, sorry. And this is asymmetric at a circumdata. Of course, this is not too widely available here, but in a nearby place near the Garu Hills, it is found, but not in.
Professor Barua,
yes.
just sorry for the interruption.
Yes.
We will be also simultaneously presenting your slides. Okay. So
okay, fine.
Okay, we'll do that. Okay, you keep talking. Yeah, you keep delivering the lecture, and simultaneously we will be. And you please stop sharing. You please stop sharing so that you are audible, and we'll be sharing the slides so that the participants can see actually what you are presenting. Okay. Yeah.
Okay. So
you can stop sharing. Yeah, we will share these slides.
Okay.
Yeah. Yes.
Okay. So, so this is our asymmetric circumdata. It is found in small numbers, of course, about 60-70 kilometers from our Guwahati University campus. So we have to take our spectrometer, that photomultiplicative oscilloscope, everything there, and set up the lab in a villager's house, we could call. Okay, this is the sclerosia substrata. It is found in a very small number, but of course there were 2 big tanks, big ponds were there. I mean, on the banks that we see this sclerosia substrata. So this is the winter species. Of course, it is found in the month of December and January only, and that too near the Assam Arunachal border. Of course, it is again in a very small number. So these are the 4 species that we have worked in India. Now, of course, as I have already told you, why study in vivo? So this is the only way to have some idea on the nature of the reaction and on the light emitting system inside the body of a live firefly. See, it is a cold system. I mean, had it emitted heat, I mean, if it had emitted the heat radiation, then maybe some information we could have got. But because this is the cold system, so we have to rely on the light and pulses only. So based on those only, we can have some conclusion or at least some hypothesis or speculation about the reaction or the system. Okay, but these are spectra, but you need not be worried because it is not physics that I am going to speak on. It is a colored spectrum. So my intention is to show that a typical that a firefly emission, it has 3 color sectors: green, yellow, it's a narrow sector, and this is red. So 3 color sectors are there. So apart from that, no more physics. So it is just the color part only, and this is for asymmetrical circumdata. Again, green, and the yellow part is of course not that intense. It is, and this is the red part. Now it is in a spectrometer. So it is a left side in A, it is the male spectrum. This is female spectrum. See, there the peak wavelength, peak. These are at approximately the same, but female peak is at a slightly lower wavelength, slightly. But then we have, because this is a molecular spectrum, so the 0.5 or 0.6 nm is nothing. So we report it to be approximately the same. But for asymmetrical circumdata, the peaks differ by about 5 nanometers. In A, it is for male. In B, it is for female. Asymmetrical circumdata. So here, you see that the peak wavelength, it differs by 5 nanometers. So it is not a negligible value. So of course, I have no idea about the other cases because nobody has done this, because females are not very easy to get, and also it is quite a painstaking one. I mean, at night, everything has to be done at night, and catching something, doing the recorder spectrum analysis. So it is not so easy. But so you can think of a major project type of thing, so that all the species on this planet, at least in India, are characterized like this, that the female emission wavelength peak is smaller than male emission wavelength peak. So it is suggested that emissions for different species are at slightly different wavelength measure. It is due to slight difference in enzyme structure. So because the female peak and the male peak are not at exactly the same wavelength, so their enzyme structures must be slightly different. So it necessitates a study on the female enzyme structure compared to the male one. Now, these are flashes from Luciola proesta, which is the widely available species here. So at 30 degrees centigrade on the left, it is for male firefly flash. It is about 97 millisecond duration. And for female, say about 190 or 195, meaning approximately double. So flashes from female fireflies are about twice as long as the male one. See, if the reaction is fast, it means that the light emission starts and finishes fast, meaning the pulses are likely to be short. If the reaction is slow, meaning light emission starts and slowly it ends, meaning that the pulses are likely to be long or broad. So we can, applying this simple logic, we can imply that this reaction lasts longer, probably proceeds slower in females than in males. Now, you zoology people, you can think of regions for a female flash lasting considerably longer duration than the males. It is left to your own, your speculations or hypotheses. But this is a fact, and in a certain Japanese species also, I can't remember now. So it has been reported that the female flashes are 2 to 5 times longer than the males. Means that the reaction goes slowly, likely to go slower in the case of females. Now, in the case of asymmetrical circumdata also, you can see that it is already split. There are 3 peaks you can see. But anyway, that total duration again is considerably longer than that of a male. So means that in the case of the female, the reaction proceeds slowly. And that is why it lasts a longer duration, which is the most probable cause. So at different temperatures, at 20 degree, 25 degree, 30 degree, 35 degree, and 40 degree centigrade. So the 5 temperatures are shown here, and you can see that this duration, that starting and finishing, it lasts, say for 20 degrees centigrade, it is about 128, 129 millisecond. At 25, you can see it is about 113, 14 millisecond. At 30 degrees centigrade, it is about 97 millisecond. At 35 degree, it is about 83, 4 millisecond. At 40, it is about 68, 69 millisecond. Meaning, with the increase in the temperature, the reaction speed is increasing. Why? Because if the reaction is fast, it starts and finishes quickly, and that's why the flashes are shorter. So that kind of a simple logic we can employ, that if the pulses are short, it means that the reactions are quick. It is quicker. So at the different temperature, you can see that the pulse duration or flash duration, it decreases, or the speed will be just a reciprocal of that. Meaning, if the pulse is of a duration more, meaning speed is slow. That's the reciprocal of that. So it will be sort of, it will be just the reverse of this. So that longer duration will mean the slower, and the shorter duration means it is the faster. So we can conclude that here, of course, it is not exactly linear, but from 25 to 40 degree, we can say it is substantially linear. But chemistry textbooks say that enzyme catalyzed reaction, the speed increases exponentially with the temperature. But okay, it is exponential, all right. But for a short range, say 25 to 40 degree, it is substantially linear. So this is what our in vivo, the flashes at different temperatures suggest. Now, at low temperature, so when the temperature is reduced, you see that a simple flash is split. 2 peaks are there, 3 peaks are there, and in the case of female, it is 3 peaks only at 17 degrees itself. Whereas for male, of course, the temperature has to be reduced much lower. Okay, about these are a table I want to skip. Just that these are all in millisecond, the tenths of a millisecond. That the lifetime means, say, it is a maximum value, and 1 by E, meaning 1 by 2.7, approximately 2.7 times is maximum value. Meaning, if it is 1, about 0.37. So if it is 1, up to 0.37. So the time required is the lifetime, meaning the average time oxylucifine stays in the excited state. So it is in the tenths of a millisecond. Okay, I want to skip this tabular form. So, okay. So luminescent decay, it shows more than one lifetime. So it will indicate more than one structural form. So because we have now obtained 3 peaks by splitting a simple flash into 3. So these will represent the 3 luminescent forms of the excited oxylucifine. So oxylucifine has 6 forms, of which 3 are considered to be luminescent, and it is an experimental demonstration that the 3 peaks appear when the temperature is reduced to a very low value. And next one, so it is an anesthetized firefly. It's the control experiment. The firefly, it inhales ethylaceted vapor. It becomes unconscious. Instead of flash, a glow appears, constant glow. It's a continuous light you can go. So by using some electronic circuitry, and this is an oscilloscope, so that continuous glow, it shows a continuous train of, you can see, microsecond duration pulses. So you can say that this is one end state you can call. So 1, 2, 3, in that reverses. Again 1, 2, 3, then one which is called an end state. Again it reverses. I mean, if you put the mirror here after 3 pulses, the next set of 3 is the just the mirror image you get. So the 3 structural forms are also evident here also, but here that lifetime is a couple of microseconds only. And what do this imply? It is a different thing altogether. So this is for another specimen. So we can say that the firefly light is the manifestation of an oscillating chemical reaction. So as if the red sector, then yellow sector, the green sector light, then one end state comes again, green, yellow, red, as if it is in a oscillating chemical reaction. It manifests. So this is what our in vivo emitted pulses suggest. Okay, so it is the last part. At what time the fireflies will appear after sunset, and why a particular species chooses a particular habitat. So can our emitted light and flashes speak something on that? Okay, we will see. Okay, so after the sunset, as we all know, the fireflies come out after sunset. But there are basically 2 broad groups. So one is early starting or dusk active, meaning those fireflies begin flashing within 30 minutes after sunset. And another, the group you can call as a dark active, which begin flashing after 30 minutes of sunset. And it is shown that it is, of course, it is at a peak wavelength. Our Luciola proesta is 562, and the Sclerosia substrata is 558. So the maximum, the wavelength peak, if it is less than or equal to 558, it is supposed to be dark active. And if it is greater than or equal to 560, those are dusk active. Right. These 2 are the broad categories. Okay, we will check. But this is generally correct, but not entirely, because there is the one paper which are about to submit where it will be shown that this asymmetric data, even though its wavelength is high, it is not an early starting one. So our Luciola proesta, it is 562 nanometer, so it comes under early starting category, meaning it is dusk active. Sclerosia substrata, its peak is 558 nanometer, so it should come under that starting category, meaning dark active. That's why it is like that. So when the temperature is increased for Luciola proesta, so this is at 27. At 42, you can see the peak is gradually shifting towards right. At 45, I said at 43, 44, all these are, I have skipped. At 45, you see it has shifted towards right. So it becomes 567 nanometer. So just after this, I mean, it is about to shift here and it has shifted to 567 nanometer, meaning there is a shift towards the higher wavelength side. So this is the lower wavelength side. and starting with, so then it goes towards the right in your screen, it is the higher wavelength side. So it is shifted towards right. So what do this suggest? What should be the chemistry or the biochemistry behind it? So you can see at 42, this is a light organ of a male firefly at normal temperature 26 degrees centigrade. 42 degrees centigrade, you can see that there are tinges of red at the border, meaning some kind of red shifting is occurring. And the 45, you can see it is that the red part is considerable here. It is a visual indication of the shift in the wavelength peak. So the pulses you can see at 30 degree, it is about 97 millisecond. It lasts here, lasting time. At 42, it becomes the minimum, about 50, 56. And then for a small increase in temperature after 42, there are considerable increase in the duration of flashes. It becomes considerably longer for small sense. So if you show it in a plot, you can see that with the increase in the temperature, this graph shows that the flash duration decreases, it becomes a minimum, and then increases considerably with large fluctuation, with small increase in temperature. So what do this suggest? So at 42 degrees centigrade, we have found that the wavelength peak, it tends to shift towards red. The pulse widths become the narrowest. So the catalyst, that the luciferase enzyme, it probably gets denatured, meaning it's there must be some that ordered nature of its structure is lost, and it is irreversible also. Meaning it is a protein, so its structure is probably some bone, maybe broken. So it is denatured. This is what we can comment that. So 42 degrees centigrade is its denatured temperature as far as our spectra and flashes say. The next part for sclerosia, you can see at 34 degrees centigrade itself. So this is for 28, this is 34. It begins to get shifted towards right. And also, this means full width at half maximum, FWHM, meaning if this is the peak, so the half maximum you take, and the width is called half width or FWHM, at half maximum. So at 45 degree, you can see that the peak is shifted considerably towards right. I mean, it is totally denatured. At 34 degrees centigrade itself, it just about it is beginning. And then you can see that 35 degrees centigrade, 37, 39, 41, 43, and 45. The peak it gets shifted towards the higher wavelength side, meaning it is called the red shift. Towards the high, it is red, and towards the low, okay, we will call it as blue. And about the pulses, you can see starting from 25, with the increase in the temperature, the pulse width or pulse duration, it becomes shorter and shorter, and at 34, it becomes a minimum. See, it started with about 185, 190 millisecond. It became about 115, 120 millisecond at 34. But after that, the next slide, from 34 degrees centigrade onwards, for small increase in temperature, you can see considerable increase in the duration time. So it becomes approximately 200 or something here. And for 1 degree increase, it becomes 300. At 45, it becomes more than 400, approximately 400 millisecond. And at 40 degree, it is about 470, 475 millisecond. So they are considerable increase. And at 43 and 45, these are no on-off type of flash, just fluctuation of intensity only. So we can say that the 34 degrees centigrade, it is the temperature at which the flash duration becomes the minimum. It means that maximum rate of the reaction, because it is fast, and that is why the duration, I mean, if it is fast, it starts and finishes fast. So a flash is likely to be short. So, and along with that, in our emitted light also showed some red shift. The peak is shifted towards right, meaning there must be some structural change in the luciferase. So, and also it is irreversible. I mean, if you lower the temperature afterward, it will not come back to the original situation again. So, next slide is the visual indication of the lantern of a male firefly. So you can see that at 34, okay, it is not too clear here. At 36, you can see that slight tinges of red come. It is 42, and at 45, you can see it is completely red. It is at 28, you can call it is a greenish yellow. At 34, that the green part is reducing. At 36, the red part is coming slow. I mean, in small amount, of course. At 39, it is a 42, no green here, and at 45, it becomes red. It is a visual indication that there is something is happening. So, next slide. We could roughly consider for sclerosia substrata above 34 degrees centigrade as high temperature, for Luciola proista above 42 degrees centigrade as high, meaning it is beyond their tolerance value, you can call. The temperature tolerance is the limit. Next slide. So our red shifting temperature, meaning the wavelength peak, it becomes of a higher wavelength. It happened at 42 degrees centigrade for Luciola proista, and at 34 degrees centigrade itself, it happened for sclerosia substrata. So from these two values itself, you can have, I mean, you can guess that why Luciola proista are early starting one. See, at this part, the maximum temperature is usually 36, 37. It rarely goes past that. So after sunset, it comes down to around 33, 34 maximum. So Luciola proista can easily come out. But see, for sclerosia substrata, it is 34 degrees. So obviously, it needs some, I mean, the cooler place. So that's why there are two large ponds here in our Guwahati University campus, and there are some large trees are there. So that the sets of those branches of the trees, and maybe that the big pond also has some role to play, that they keep themselves cool and come out only when the temperature becomes tolerable for them. So about the last 5 years of observation, it says that it has to be about 30 degrees approximately or less if it is to come out. So that is why it becomes late starting species. Okay, next slide. So there are some rainy days when the temperature at this place becomes approximately 30 degrees centigrade or less, the maximum one. So in those days, it has been observed that sclerosia substrata specimens of course 1 or 2, it is just about half an hour. I mean, approximately half an hour after sunset. Means that it almost becomes an early starting one. And there is a little to differentiate the appearance times between sclerosia substrata and Luciola substrata. But so a general conclusion is that majority of Luciola firefly, Luciola proista fireflies come out within half an hour after sunset on any day, whether it is hot or cold, it doesn't matter, because at our part, the temperature never reaches 40 degrees centigrade, so it is no problem for Luciola proista to come out immediately after sunset. But for sclerosia substrata, the temperature has to be, well, our observation of 5 years, it say that it has to be about 30 degrees centigrade approximately or less if it is to come out. In fact, during those COVID lockdown period about 2 years back, I went out of my home, I mean, every day, because it is at the campus, so we can go out inside the campus. So I observed that 2 particular days when the temperature, maximum temperature became 39 degrees centigrade. Even after 3.5 hours of sunset, not a single specimen of sclerosia substrata came out, because the temperature at that time was still about 31, 32 degrees centigrade. So temperature has to cool down. The next slide. So we can conclude that peak wavelength, as that science paper that indicated, it generally determines active time, all right, but does not determine its time of coming out. The temperature indicating luciferase denaturation. It is the shift deciding factor. I mean, that spectrum where the peak is shifted towards right, or the pulses become abnormally, I mean, that it becomes, it becomes the minimum and then increases for small increase in temperature. So that is the temperature. So that determines it. So next slide. So you can see that when the temperature is reduced, it becomes exponential, all right. So these chemistry textbooks are okay, fine. So I am not going to dispute that. But for a small range from 25 to, here also from about 23 up to 33, 34, it is approximately linear. The linearity is lost about, say, 23 or 24 degrees centigrade, something like this. And at 34, you can see that figure on the right, the minimum one at 34 degrees centigrade, and then it increases for small increase in temperature with large fluctuation. Those bars means that these are deviation from the average value. So it is this temperature value is where we are interested. Okay, so next slide. So as it is already shown that if you have a close look at that graph, that figure, so from 34 to 25 degree, it is approximately linear, and then it becomes exponential type of thing, meaning the linearity is lost below 25 degrees centigrade. But for Lucifer Priesta, from 23 up to 40 degree, it becomes substantially linear, and then below 23, it sharply increases, meaning it no longer becomes linear. So next slide. So our speculation is that the loss of linearity means that it is longer lasting, because the pulses or flashes become abnormally broad, meaning the reaction becomes very, very slow. So that the forces the firefly to stop flashing at temperatures lower than this. So at a low temperature, if you have a look, it is just the opposite of that, left-hand side. The next slide, of course. Here you can see that at about 11 degrees centigrade, that the second curve from below, from the lowest one, you can see that peak is shifted towards left, not right, just the opposite. 8 degrees centigrade opposite is clear. And the figure on the right for sclerosia substrata at about 10.5 degrees centigrade. I mean that the figure, it is third from the top. So you can see it is slight shifting towards left has started. And at the lowest one, 6.5 and the 7.8, it is quite clear that it has shifted towards left, meaning it is the blue shift you can call. 558 nm has become 553 nm. Okay, so next slide. So indeed, a lantern or light organ, I mean, it is a visual indication that at 9 degrees centigrade or even at 11 degrees centigrade, you can see for Lucifer Priesta, the tinges of green appear. Just a simple photograph, which becomes considerable, that the greenish part at 9 degrees centigrade. And the lower one for sclerosia substrata, you can see at 10.5 degrees centigrade, the greenish tinge it appears. And at 6.5 degrees centigrade, it has become considerable. So it is a visual indication that color is changing. The next figure, sorry, next slide. So this is the left side. It is our for Luciola proesta between the two peak shifting temperature, one at 42, the high one, and the low at 11 degrees centigrade. And for Sclerosia substrata, the high, okay, it is from 20 degrees itself that we have done, because the other one has already been shown, the 20 degree, and you can see that at about 9.5, sorry, at about 9 degrees centigrade, the fluctuation, I mean that the deviation from the mean position, it becomes noticeable. So a single temperature, it is difficult to fix. So a small range we can suggest that it is the range at which the peak shifts towards the lower wavelength side, or blue shifting we can call. The next slide. So the range we can suggest is 11.5 to 11 for Luciola proesta. That it is cold in this region, because the theory is that if there are some difference in the enzyme structure, the peak position is slightly different. And for Sclerosia substrata, it is 10.5 to 9 degrees centigrade. And it is reversible, meaning if your temperature is gradually increased, the Firefly recovers and retains the earlier characteristics. So the last part, that these the peak shifting temperatures give the reason for their choice of habitat. So why a particular species chooses or selects a certain type of habitat. Next slide. So as already mentioned that normally at this part of the globe where this Sclerosia substrata and the Luciola proesta those dwell, it becomes the maximum is around 36, 37 degrees centigrade usually, but sometimes it reaches even 38, 39 also maybe in a day or two. So the shades from branches and leaves of trees in the banks of the big ponds, it gives the temperature within the optimum temperature, meaning the peak shifting temperature of Sclerosia substrata. So in the hot days, these fireflies start flashing sometimes more than 3 hours after sunset. So they have to keep cool, otherwise it will be intolerable. And next slide. So this is the place where Sclerosia substratas are collected. So it is a big pond, and the right side of the small road is also, there is a big pond there, but it is not visible here. So it's at a very big pond. So in these trees, the Sclerosia substrata are found. The next slide. So there are the blue shifting temperatures of Luciola proesta and Sclerosia substrata are approximately the same. So they disappear at about the same time at the onset of the winter. So why Sclerosia substrata also chooses this particular locality is that because there, of course, the temperature could be a little bit higher than they would have liked. But okay, because of these big ponds and the shades of the trees are there, those keep them cool. And about the winter part, of course, the blue shift occurs at about 10.5 to 9 degrees centigrade. So they disappear at about, I think, towards the end of October. In this place, they disappear. So just the last slide. Our conclusion is that the red shifting temperature, it determines whether a species is dusk active or dark active. The next conclusion is that the blue shifting temperature, it gives the reason of disappearance of a summer species just before the winter sets in. And finally, these two temperatures, and of course, the food has to be available. That's the foregone conclusion, of course, decide the selection of a habitat for a species. Okay, thank you for your patience.
Yeah, thank you very much, Professor Barwa. So it seems that the entire activity of fireflies is temperature regulated, and the bioluminescence and the properties of light are all governed by the temperature. What may be the role of humidity or moisture in this, Professor Barwa?
Okay, the humidity and the moisture we have, whatever little we have checked, it has got hardly any effect. So we have not found any effect. Of course, some detailed study need to be performed, but it is the temperature studies are easier because I mean it is instantaneous.
Okay.
I mean at Japan, when I did some magnetic field study,
Yeah.
there I found that it took a long time for some change to occur. But with the temperature, it is instantaneous. I mean, the moment you increase the temperature, the pulses become shorter. Or the moment you decrease, lower the temperature, the pulses become longer. So it means that the reaction is, the speed of the reaction is affected. If the speed is fast, meaning the pulse will be short.
Correct.
So that is how we have concluded about the speed of the reaction. Otherwise, you will have to kill the insect. And that way the real situation will not be there. So you have to extract the luciferin and luciferase and carry out your study in a test tube maybe, but that is an in vitro one, and that may be a controlled one, but that is not the real situation. The real system which is made by God. So inside the body of the system, some reaction is going on which emits light. So after analyzing of those light and flashes, so we have made some conclusions about the nature of the light emitting reaction or the light emitting system inside the body of a live firefly. But this is what is our objective. And of course, yeah, you cannot say that it is conclusive because see, we have only studied only the emitted the flashes only and the light only. So to conclude that it is denatured, so of course, I mean, you have to carry out some thermodynamic studies that the chemistry people are experts there, and there are 4 parameters you will have to determine. Only after that you can conclude that yes, it is denatured. Otherwise, ours is only suggestive only. It is not exactly conclusive, but almost conclusive you can call.
That we need to have a multidisciplinary team, isn't it?
Yes.
Yeah, we need a physicist, we need a biologist, then
Yeah,
we
definitely.
need a chemist. Yeah,
Yes,
we need
yes,
a
yes.
chemist.
Yes,
And
yes,
so that we can have a multidisciplinary team, and
Yes.
we will per se or in combination investigate the process of bioluminescence.
Yes,
No doubt, Professor Barwa, your studies have clearly shown that most of the properties of bioluminescence in fireflies are related to temperature. Now, have you got any other reference where under aquatic systems or ecosystems, how these fireflies use the photo radiation, especially for communication or for completing the biology? Any hint or idea?
No. Actually, in that area, I had not studied. But see that female flashes, no? That are females also flash.
Yeah.
I mean, in response to the male flash,
Yeah.
females also flash. But female flashes are considerably longer. It is more than twice as broad as in the case of male.
Yeah.
So, I mean, you can make your own hypothesis of why the female flashes are so large compared to the male. That longer flash means that the reaction, I mean, as far as the reaction is concerned, it has to be slower.
Yeah.
Or a longer lasting reaction. So in the case of that, because this is a mating signal, so you can have your own hypothesis or speculation that why that the nature has created this, that the female system in such a way that their flashes are considerably longer than the males. There must be a reason, because the mother nature does not do anything casually. There must be a reason. So arbitrarily she will not do anything. So there must be a reason. So okay, you can have your own conclusion there.
Yeah. But could you get down to the question as to what exactly triggers bioluminescence in fireflies? So we know that there is some kind of rhythmicity, or there is some kind of what you call activity, pattern of activity. But what actually think is the mechanism of trigger in the fireflies of bioluminescence?
There is a hypothesis that a kind of a nerve pulse, which is called octopamine. Octopamine is supposed to trigger this bioluminescence reaction. So it is a nerve pulse, you can call. But well, as you all know, oxygen is the trigger. But it is supposed to be initiated by a nerve pulse, which is called octopamine. So it is, of course, it depends on the neural condition, of course, because if a firefly, suppose if you stir this, the flashes will be irregular time. I mean, so obviously some nervous system has been affected if you stir this or if you scare it. So the flashes will not be regular. So whatever flashes I have shown is after making it acclimatize and waiting for a considerably long time, so that the flashes become uniform. Only after that, those recordings in an oscilloscope will done.
Yeah.
So
No.
those things.
Yeah. One of the researchers, Ms. Devi Priya, is asking whether there is a definite period for fireflies to emit light. There is any
well, the fireflies emit light, of course, after darkness, because we have tried in the daytime also after getting the firefly. But no, I mean, if you stir this, maybe for a couple of seconds, it might flash, but those are irregular ones, and it is, I mean, they do not flash during the daytime. It is only after the sunset. So, well, I have seen that up to midnight, even up to the late night also I have seen, but mostly about the Luciola proesta, about 10 to 11 o'clock, most of those are seen. After that, it decreases in number, flashing fireflies.
Okay. Yeah. Any other questions from participants? Participants, would you like to have some questions with Professor Barua? They can directly ask. Yeah.
One thing I just want to apologize that I tried to be as simple as possible, because as little physics as possible. Yeah. But then if it becomes awkward for you, I apologize for that. Because
no, not at all.
So it will mean that I have failed.
No, not at all. It has been very interesting, Professor Barrow, so to say. And in the beginning only, we said that it's quite a complex phenomena, and to dwell on such topics as this would be very difficult on single-handedly. And that's why we need to have a multidisciplinary team.
Yes. And also, I would like to suggest that you may think of starting a kind of a project, at least in Indian context, where all the species of fireflies are characterized. All the species, each and everyone.
Sure. With your cooperation and guidance, surely I think we can take this.
Oh, your help will be definitely needed.
Yes, we will be there. We will be there. And another interesting question is, sir, do you have any observation on Indian fireflies directly related to water bodies? This is a question from Dr. Bijoy.
Oh, the water bodies, well, I do not know, because what actually we have done is that we go out and catch the fireflies and bring it to the lab and do our research. But well, we make some observation also regarding their appearance time, at what time they start flashing after the sunset, what is the optimum temperature for them. I mean, that comfortable temperature for them. But about the relation to water bodies, well, sorry, I will not be able to give you the answer.
Okay, okay. So I think we will wrap up this session, very interesting session on bioluminescence in fireflies, Professor Barwa. And we thank you for your effort, time and energy for having spent and informed the researchers about the properties of bioluminescence in fireflies. We definitely would like to have more active interaction with you, and we will also be circulating your PPT to all the participants, so that if they have any more questions, and we can also see more questions are coming, so we will direct all the questions to you, and participants will have an opportunity to have your feedback. Thank you so much, and have a nice day.
Thank you very much.
Yeah. Yes. Ladies and gentlemen, now we just now heard Professor A. Gohain Barwa, and now it's the time for the participants, the young minds, and the youngsters and the researchers who would like to share their experiences. And we have with us expert, and we are very fortunate that we have with us Professor Leslie Ballantyne also. And others. So definitely, if you have would like to share some experiences, you can do so now. I hand over this session to Mr. Parvez, who will be coordinating this session. Now, after this session is over, we'll have a tea break for 10 minutes, and then we will move on to the conservation of fireflies. And we have with us an expert in Professor Sarah Lewis, who will be sharing her experiences and coordination activities for conservation of fireflies. This will be followed by a lecture on fireflies in Malaysia by Dr. Nada. So these are the upcoming lectures, so please wait for the time. Meantime, let's hear to our young minds. Over to Mr. Parvez.
So good afternoon, everyone. So this is Parvez. I'm working on fireflies. I hope you must be enjoying this session, and you enjoying the session and the webinar and enriching yourself with the information, these puzzling beetles. To know what we know about fireflies, we have developed questions for like participants, like basic questions, 20 questions on fireflies. Mainly focus on to understand basic about fireflies, like among the participants, like what are the basics of fireflies, how, like from which family, from what are the background of fireflies. So we are about to share the links of like a quiz. So please participate. And the chat box, you can find the link, and please do participate. And simultaneously, if somebody wants to share their experience on fireflies, it's more welcome. Mr. Arpan is there.
Arpan Kushti. It's going to unmute. Yes.
Hello.
Yeah.
Yes,
Hello.
you are audible.
Yeah.
Yeah. Okay. Can I please? Am I visible?
Yes.
Okay,
Yeah.
okay.
You're visible.
Can I please share my screen?
Yeah.
Hello.
Yes,
Hello.
you are visible.
Okay.
Yeah.
screen. Okay, is my screen visible?
Yes.
Okay, so first of all, thank you so much, Chakravarti sir and Parvezda, for this opportunity. Like, I am greatly like interested in fireflies, and to be able to share my work in the presence of Professor Ballantyne and Professor Lewis, like they are like my greatest inspiration. So I'm very, very honored for this opportunity. So I would just like to share my little experience that I have done during my master's thesis. I chose to work on the species Abscondita, the genera Abscondita, and I constructed something which was known as the Firefly tube. And in my experiment, I wanted to study how artificial light at night can actually affect the distribution pattern of these insects. And also a very interesting observation that I had made in my experiment, which was food, which can act as a masking factor in their distribution pattern. So are the slides visible? Hello?
Yes, 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 yes. It's visible.
So all the scientists have always said that light is something that is like the reason for their decline of these species. But when it comes to photo-induced movement, we see that these insects with light pollution is something that affects them, but they are again attracted towards something that is actually going to kill them. So that was the question in my mind that I went up to my sir and said that I would like to address what is the basis behind this potentially lethal attraction in these insects, and how would these species respond, and also whether food can act as a potential dictating factor in changing this preference over time. So this was my experimental design, where I constructed a firefly tube, which was about 150 centimeters long and 25 centimeters in height and width. And I introduced dilemmic situations on both ends of my setup, like light and darkness, and like food and no food, and also like established a light gradient along which the fireflies dispersal pattern would be observed for a period of 6 hours, ranging from sample sizes 1, 5, 10, 20, 40, and for 6 trials of each sample sizes. And this was the actual setup. It was very simple. It was just made of cardboard and painted black, and there was like light sources on both the ends, which was measured using a lux meter. And during my uh, uh, project, I could see that I could like, with a lot of like video evidences and a lot of graphical representation, I could see that the fireflies were greatly attracted towards light. But as soon as and also we also switched the light sources and saw that the attraction was towards the light. Like in the video, you can see that the fireflies activity in the lit area, which is the red area of my firefly tube, increased manifolds compared to the blue or the greener area, which are actually in the darkness. So the fireflies were attracted towards light, which was expected. But as soon as I introduced food into my setup, the preference actually reversed, and the fireflies were now attracted towards darkness, irrespective of whether only because the food was present there. So when it comes to, so what were the conclusions that I made from this experiment was that when it comes to the population picture, I think food, which acts as a proxy for the vegetation or the habitat of the fireflies, can actually work synergistically with light pollution, and should be given equal importance when it comes to conservation related studies and such studies to understand how the distribution pattern of adult fireflies changes with time. So and also like in the population, we can say that artificial light is actually responsible for changing the feeding duration and also the vegetation growth. And on the flip side, vegetation can be masking the effect of light. So artificial light at night and food are synergistically working towards affecting these species. And in the future, I would like to continue working on fireflies, and also understand like how different types of light of different wavelength and different types of food can actually change the distribution pattern of both adults and larvae. So yeah, this was my little work. Hello. Hello.
Hello. Yes. Yes, Arvind. Thank you for sharing your work.
Yeah, like, did you, was I audible and visible?
Yes, you are.
Okay. So yeah, that was my work, what I have done for my master's thesis.
Thank you so much, Arvind, for like sharing your work among participants. If anyone have to share your like experience about like fireflies. Yes, ma'am, please.
Okay, Arpan, you said that this was Abscondita chinensis, I think you thought it was, but you said it fed. What was it feeding on? I think he's muted, Mr. Parvez. I think he's muted himself again.
Oh, hello.
Okay,
Ma'am,
right.
first of all, I am like, I can't really believe that I'm talking to you right now. Like, you are one of my greatest inspiration when it comes to,
Golly,
so like,
you've
you
made
are
my day.
like, it's like a dream right now. So yeah, I'm like hyperventilating a little bit. Yeah, it was Abscondita chinensis, and actually like it was identified from your work on Abscondita, the keys that you had provided in one of your papers. And regarding what was it feeding on? Well, first it was, that is the thing, like most fireflies do not have like, they are supposed to have degenerate mouth parts, but
No. They
like,
don't have degenerate mouth parts. They have very well developed mouth parts. They
yeah,
may just not feed as adults because there isn't very much time.
but the adults are actually like, they usually do not feed. That is what I guess I have heard. So, but interestingly, what I saw during my when I was like keeping them and culturing them, I saw that they were actually very attracted towards apple, like extremely attracted. So now what I did, like I told my sir said that like you have to like establish like a scientific relationship. So we did our experiments with the help of 60% sucrose solution in cotton. And but yeah, that is what came to my mind, that if they are feeding on, I'm not sure if they are feeding on the apple, but they are definitely attracted towards it.
It may just be that they're taking in some fluid, because they have been observed on leaves to take, if there's a little droplets of water in the morning there, that they will actually take in a little bit of fluid. So
yeah, that is what is interesting. I would like to like continue on working on whether different kinds of food can actually mask the effect of this lethal attraction towards light in these species. Like,
I think it's wonderful.
Thank you so much for inspiring us.
Oh gosh.
Now we have one of a participant from Atri, Miss Ranjini. She want to share some like experience. Ranjini, over to you.
I just wanted to share the kind of a small initiative which I started last year to well, towards trying to establish like a baseline data about fireflies in India, like, and what's happening to their population, stuff like that. It was just an idea. And then I just created this simple form that I circulated with people in different mailing groups. And the form collected information on when did they sight fireflies, how many fireflies they sighted, along with the location details. So right now I have like information from 150 sightings across India, like a location map on a map of India. I have like little spots from where people have sighted fireflies, how many they sighted, and what kind of habitat in which they sighted them. So I was just kind of wondering how this kind of information might be useful. Like, I just started it, and then I realized now I have information on firefly sightings, but obviously we don't have any information on what species they were. And this is just the second year that I'm continuing this work and trying to get more and more information from across the country. So I just wanted some comments as to how this could be made useful in a proper monitoring of these species, was one kind of a question. And then the second one is the flash patterns. Like, I'm hearing that with the flash pattern we can maybe try and identify the species. So I was wondering, like, could I ask participants to record like a 1 minute video or a half a minute video of fireflies, and from those flashes would we be able to get any useful information? So yeah, these are some questions that I had.
Mr. Judy, it has been a very good initiative, and certainly it's an effort that we are also at it. Okay.
What's happened?
So it is hanging.
Yeah, I'm unable to hear.
Ranjini, can you hear me? This is
Yes.
Leslie.
Yes, I can hear you, Leslie.
Oh, okay. Well, I hate to say this, but you really need to go back to all those sites and try to collect some fireflies. Now, you may not necessarily go back to each one, but is it possible that you could get somebody from those different sites to collect some for you? Now, I don't know what sort of liquid they could put them into, but they should immerse them in something like, I'd suggest methylated spirits. They're not going to have 70% alcohol or anything like that, but that will keep them nice and soft, and you'll be able to see them in their fairly natural state. But it's, you really, the next step is, as you said, is you have to be able to identify them. So that was your first question, at any rate, I think.
Yes, great. Thank you, Leslie. I mean, I did realize then I've been finding out what are the rules and regulations around collecting. And I'm kind of finding that the fireflies are not coming under the scheduled insects. And if they are found outside the protected area, I think it's all right to collect them, is what I've kind of understood so far, which is also something I would like to ask the group here. And if that is the case, I'm trying to contact these people who recorded their details in to ask, can they send me specimens? That's something I'm trying to do so that I can start, as you said, identifying who's the species present where and things like that.
Yeah, I think it's a fantastic project. Very, very interesting to sort of get that sort of information, and just be very nice to all the people who send you specimens.
Yes, I will. Thank you. Could you also comment on the flash pattern, Leslie? Will it make sense to ask them to collect record videos? Will that be informative at all?
Yes, there's a limit. The thing is, flash patterns should theoretically be recorded with a very high resolution camera, and you do need to record the temperature. So flash patterns, I mean, even from what Dr. Barua said, it is clear that flash patterns vary depending on what the temperature is. But I think that there's a lot of potential there. Now, I know that it is possible. I've got an iPhone here, and I know that it is possible to video fireflies on an iPhone. It's not the greatest, but it's not bad either. And if you were to get in touch with me, I think I could possibly hunt up some references for you of a program that will translate the flash patterns on the video into a graph that could be quite useful for you. So if you just look up my site, you'll find my email there somewhere.
Great. I will certainly do that. Thanks a lot. I mean, even if not in this kind of an initiative, I'm thinking of trying to start something with the forest department in some places. So maybe they could be trained to take these videos and stuff with good instruments. We could try and do that also.
Yeah, well, you know, the thing is that I know that the high resolution cameras that you saw in my talk when I at the very beginning, I had those people were taking our Blue Mountains Firefly. They're pretty expensive cameras, but you can do something with an iPhone too, with not necessarily an iPhone, but a phone that's got you can take a video with it.
Great, that's very hopeful. Thanks.
Okay.
Yeah. Any other participant would like to share?
Well, can we take a sort of a suggestion? Can you put for the selected pockets should be kept dark?
Yeah.
Because in our campus itself,
Yeah.
I think about 2-3 years back, I also made an appeal to our Guwahati University campus dwellers that the selected pockets should be kept dark, but nobody hit it. So because of this light pollution, that is what Dr. Valentine said in the morning. Because of this, so the fireflies are leaving some certain locality because of this light. So I mean, there's some kind of not exactly the policy. The steps should be taken to make selected the pockets should be kept dark. Otherwise, the preservation of fireflies will be difficult if too much of lighting is there.
Yes, Professor Barrow, I agree with you.
Yes,
but practically, the thing is that we cannot impose this in all areas. Maybe in select areas we can do so.
Yes,
and
yes, yes.
In protected parks, national parks, wildlife sanctuaries, and wherever we are having hotspots of fireflies. Definitely, this light menace or pollution, what we can say, can be regulated or controlled very much so that their deal activities are not affected.
Yes, yes.
Yeah, Madam, I wanted to ask Professor Leslie Valentine that how we can attract the school-going children. Because in India, what we have seen is that the moment they see a firefly flashing, they would be very much enthusiastic. They will collect it and they will sort of destroy it. And they will also try to play with its flashes. So any such initiative being done across the world or in Australia?
No, not in Australia, because we don't have a lot of fireflies, but they're not really in association with cities or even country towns. So what I suggest you do is when Sarah Lewis comes on, ask her that question, because in the USA, fireflies are in everybody's backyard almost. You know, they grow up with them, and I'm sure that it would be possible to you know, instill a culture of perhaps a little bit more care.
Yeah. Yes. Yeah, that would be very interesting, and that's how we can make advances in understanding fireflies. Yeah. Any other questions from youngsters, especially? And I think we are also going to conduct a quiz. And, you have already sent the forms, and youngsters and researchers are requested to please fill it up. So we would like to know that how far what we know about fireflies. So this will help us in designing programs for firefly conservation in future course. I think What's it? Just for 17. Any other questions from the participants? Then Ranjini, what are the other initiatives ATRI is taking on fireflies? Any other initiative apart from sort of counting or reporting the emergence of adults? Any other initiative the ATRI is taking up? Ranjini?
To whom did you address that question?
Yeah. Pardon?
To me?
Didn't address the question. Repeat.
The thing is that apart from the records of the flashing fireflies, any other initiative being associated with this activity, I wanted to know.
Well, the whole thing is that they need to know where the larvae are living.
Yeah.
Because the larva is going to determine,
Yes.
as I said, when.
Yes.
So I think if you could, it's usually a matter of sieving through, using Belase funnels and sieving through the leaf litter to find some larvae.
Yeah.
So you could determine what sort of larvae they are, and that would give you some notion of, if they're living in leaf litter, then you've got a pretty good idea of what their environment is like. So I think that what Ranjini was talking about was, if she could make some sort of record of the environments that she's getting all these records of adults from. It's a very exciting project she's got going there, I think.
Yes.
Yeah, yes. So this is not exactly A3's project. I'm a PhD student at A3, but this is a project that I got a small fellowship from Earthwatch Institute for a year to do, and this is I've just started collecting these information. Apart from that, we have not done any kind of ecology related work yet on fireflies, which I hope to do in the future. But we do have Leslie, like a basic information on whether it's a plantation or a paddy field or a built up area or where people have spotted these insects from. So we kind of have an idea now whether the majority the larger sightings are from forests or not. Such kind of a basic idea starting to emerge.
Okay, so you can immediately eliminate whether they're going to be aquatic.
Yeah. Yes, that would be very interesting. Okay. Dr. Gupta, would you like to say something as far as the popularization of fireflies in the right direction in India? Because you have been associated with beetles for a very long time, and with your vast experience, you
Sir, you are not audible, sir.
Am I audible now?
Yes, sir. Yes.
Dr. Gupta, I was telling, see, the fireflies have remained very much apart from the public and especially the young researchers. Would you like to suggest some initiatives which we can take on a national basis and propose some kind of project so as to make these beetles and like the jewel beetles, the click beetles more popular, and so that the youngsters will develop a proper perspective of
Sir, thank you for this popularization of this we can do. What I suggest that we can come out with a field guide of fireflies of India. We have very few species, and Dr. Valentine Madam is here. We can take her help, and if something can be brought out, and I can help also, because somehow I am dealing with in charge of publication also. So I can somehow got it published. If we can come out of this conference or webinar, if some field guide of fireflies of India, In a very,
Yeah,
madam,
great idea.
Yeah.
in a very means very small rupees, like 300 rupees, very small amount of money. Then it can be distributed, or children can purchase. Even the PhDs or many people can purchase. We have 45 to 50 species in India, and I can work out on my collection and your collection. Everybody, we can collaborate together and come out with a field guide with a proper detailed distribution map of the species, with a good photograph of the species. Then we can popularize fireflies among students, college students, all those people. We can popularize them. And in this, for publication, I may be of some help. That from ZSI we can publish that publication. I can discuss with my director. If she agrees, then we can we can take the responsibility of that. We can together work on that.
I think that's a wonderful idea.
Thank you, ma'am. Thank you. All the species account photographs, we include it, then it will be very, like, the many people are asking that which is with this species, which is this species. Many youngsters are asking that. For that it takes so much expertise. If we can, field guide will be there, but madam, without your help it won't be possible.
It may well be appropriate to have a look at the sort of thing they've done in Taiwan, because they have a similar sort of thing. They're very oriented towards their, and it's just a little folder. They just fold it up, so it's a piece of paper that's folded over a couple of times. Nice glossy paper, beautiful pictures, very colourful, and absolutely excellent idea.
Thank you, ma'am.
I wish I'd thought of it myself.
And some pamphlets also we can generate, like
Yes.
Well, especially pamphlets could be geographically oriented too, couldn't they, to the species that were found in that particular area? Yeah.
And I suggest Dr. Chakravarti sir to, if a portal can be made, like many people are recording their photographs, videos, all those things, and that can be submitted.
Okay.
That sir, you initiate from your point. That will be best, because you have initiated this in Western Ghats. Then we can start with all over India.
Okay.
That you can submit the videos, photographs to you, and on one portal.
Yeah.
And also I will suggest that through this webinar, we can come out with a group, Firefly group in India, taxonomy group.
Yes.
Like Google group type thing, so that we can share our literature, our data, all those things among each other, all the participants of this webinar in one particular taxonomic group.
Yes.
Google group. And Parvez can take
yes, sure.
can take that thing, take all the emails he is having, everything. He can come out with a Google group. Then
we will do that, sir. This can be done, and it's a very practical solution and suggestion what you have given. Certainly, we'll start with the portal, the group, and we are also coming out with a small flyer, as Professor Belton has put. It is geographically oriented towards a species in Western Ghats, and where millions of these fireflies emerge during September, October. So we are waiting for their this time, and definitely we are going to make a big dent, and we'll also try to declare that site as protected site. So these are some of our plans, and we hope we will successfully be able to execute this. Yeah. Any other young researcher? Any comments or experience from them? You're most welcome. So,
Exactly. Can you say about like what we are doing?
yes. Parvez would like to share for 5 minutes his experience in rearing the fireflies. Yeah. Yes. Mr.
So
Parvez.
2 months before we have collected some like fireflies adult, and we kept Hello, is it audible?
Yeah, the screen froze, Parvez.
Yeah. Now you can.
Now, I'd start all over again if I was you.
Yeah.
Yes, am I audible?
Yes.
So 2 months before I've collected fireflies, and we have given a like a naturalistic enclosure. And after like two few days, but after a week, we got eggs. And with the recent literature or Martin et al. 2022, we are rearing them. And after 60 days, we got one larvae.
The eggs hatched.
Eggs hatched. And the first egg hatched on 14th July. So till now we have 14 live larvae, new hatchlings. So we are providing natural enclosure, and we are providing the food like earthworm, pieces of earthworm.
Snails.
So they are like feeding and giving a like humidity, giving little humidity, and for the survival. And now they are like going like live. They're like doing good and.
Mm.
So this is my experience. I have collected larvae also, but this is a first experience to rearing. As I learn about a lot of literatures, the larvae hatchling period is 1 to 2 months, 1 to 2 weeks only. But my larvae hatch around 60 days, first hatchling. So it's started.
What species? What species is it?
Maybe absconded.
It's abscondita
Maybe.
genus. We couldn't
Okay. Maybe the monsoon firefly.
Yes, ma'am.
Yeah.
We have collected.
Yeah.
That's good, because that's the only way. This is the only way you can reliably associate the larva with the adult, is to breed them through.
Yes.
That's perfect. Perfect.
Yes.
I have one question. We are preserving also. So can we preserve larvae in 70% ethanol or 100% absolute?
No, 70% is all right.
But
Yeah.
can it be good for molecular work also? 70%?
No.
Then
No,
we
you have to. You
100%.
have to. If you want to do molecular work, I think you have to collect them into about 95%.
Yes.
I'll find out for you if you send me an email. Remind me that I've told you I would look this up for you, and yeah, about 95%. So it's sort of straight into that. They're killed, fixed, and preserved immediately in the ethanol.
But ma'am, high concentration may change the morphology also sometime.
I'm sorry, I didn't quite catch that. Would you mind repeating?
Absolute, if we give preservation in absolute ethanol, 95, so it may change the color also of the species.
Well, it depends if what you want to do with them. If you want to do molecular studies with them, then you have to preserve them. But if you want to preserve the color, then you have some in 70%. So 70% shouldn't destroy the color too much, depending, of course, on what the color is. Most of the color patterns in insects aren't destroyed by putting them into ethanol.
Okay, ma'am. So it's better
You
to
don't
separate
look
them.
like you agree.
So it's better to separate them. For molecular work, we can put in the 95%. And
Yes, exactly. Yeah. You just have to, you just keep a few. I realize that there are restrictions on the number of specimens that you can take in the field. it's the same for Damakowijikun in Sri Lanka as well. That, you know, for me, well, I haven't been out collecting for quite a while now, but yeah, it's just, you have to balance all these things.
So, ma'am, one more thing, like it's a common phenomenon, like male has male and female can be differentiated morphologically through lanterns.
Now, say that they communicate, or you can tell the difference between them.
Can we identify on the behalf of their lantern, like male and female?
Okay, no.
Number of lanterns.
Unfortunately, the taxonomy of the Luciolini is still male-based. That you notice that in my paper, all the keys we had were keys to males, very, very little on females. I am trying now to write a paper. I've got quite a large paper written on the use of females, but it doesn't always work, particularly in things like your monsoon firefly. There are lots of different species that look like that. It's not just that species in India. And while we could certainly determine that a female was a particular genus, we just don't have what it takes yet to determine what species it might be. And the lantern is not the bit you look at. What you've got to do is dissect out the female reproductive system.
Yes, ma'am.
So I didn't go into that at all, but we do a lot of dissection of genitalia and things like that with fireflies.
Okay.
Okay, ma'am.
Yes, so that will be a very good indication for us to take the taxonomy of fireflies forward. I think, ladies and gentlemen, we'll have a break for about 5 minutes before Professor Sarah Lois gets in, because she's available to us only after 4:45 IST. So just we'll enjoy a small cup of tea, and then again we'll resume the webinar. Thank you so much.
Thank you.
Thank you.
Oh, really? You're in bed, right?
हाँ क्या बता रहे?
अभी वो आपके बैग में अपॉइंटमेंट है ना?
वो भेज दिया, ईमेल पे अटैच कर दिया।
हाँ?
चेक योर ईमेल, चेक योर ईमेल।
अच्छा।
Well, Sarah, it's Sarah on next. So. Hey? Okay. As I said, there's been ups and downs. The screen keeps freezing, and Dr. Baru's talk was just impossible. But we should get a transcript, or we should get at least we all get something. Right, you know. And what's his name? Chakrabarti. It's you know, it's good on him.
I just wonder what we know about using things fast. She's got a lot of work in terms of getting these people involved. You know, she had
Louis? Louis?
yeah, Lynn had that guy. The other one. Deacon that.
No, Rich David Attenborough.
Yeah.
Well, the whole film company came out.
Yeah. She like it didn't go to the video. I got a great day.
Okay.
Yeah. Yes. Good evening. Welcome, Professor Sarah Lewis, to this webinar, and we have had a very interesting and useful time in the morning and just after that, and we very eagerly waiting for your lecture on firefly conservation. Now, Professor Sarah Lewis is an ecologist and professor of biology at Tufts University. She has spent past 30 years studying insect behavior, ecology, scientific articles like the one for Scientific American, Undark, CNN, The Guardian, and National Wildlife. Her work has been featured in New York Times, Wall Street Journal, Washington Post, and BBC, along with numerous radio shows and postcards. Professor Lewis is also the author of Silent Sparks, The Wondrous World of Fireflies, an entertaining and highly accessible journey into the luminous lives of these cherished insects. Professor Sarah Lewis, when contacted, kindly consented to share her efforts and experience in ecotourism and conservation of firefly, the threats and also the other opportunities and challenges that Professor Sarah Lewis, please. Yes, madam. We'll kindly request you to begin your lecture. Am I audible? Hello? Am I audible? Oh, no.
Much better. You can hear me now?
Yes.
Okay, that's great.
Thank you.
Sorry about that. No matter how many times I do these Zoom talks, it's something always seems to go wrong. So if you could also let me know, I'm going to share my slideshow. And no, that's not exactly what I want to do. Let me just. Okay. Might just take 1 minute to
Sure.
get the PowerPoint behaving properly. Let's see if I can do that. Interesting. Okay, let's see. Share screen. See if this works. Maybe a little bit better. Sorry, my PowerPoint is acting up a little bit. Let me see if I can. For some reason it keeps closing the application. I'm not sure why. there's a recent Zoom upgrade. Let me try this. Might work. So exciting. Huh. Okay. Let's see. PowerPoint. Okay.
Yeah.
Getting
Yeah,
very
everything
close.
is. Yeah. Okay.
Um,
Don't say hello, Sarah. I think I'll just mute myself.
Hi, Sarah.
let's see.
Any problem, madam? Any problem?
Ready?
Any issue?
Yeah. Let me actually simply a problem with PowerPoint. Okay.
We can share notes later.
But she has
Let's try this. Okay, so now you can presumably see my screen, is that correct? So can you see my screen now? Or do you see my presenter notes?
Yes, ma'am.
Yes. Yes, sir. We are able to see. Yeah.
See the, do you see my slides?
Yes. Firefly conservation hopes and challenges.
Okay, sounds good.
Yes.
So,
Yeah.
sorry about that. Good afternoon, everyone. I'm very, very happy to be here, and thank you so much for inviting me to participate in this webinar. I'm actually really very honored to be here today to talk with you about the conservation of fireflies, some hopes and some challenges. So please let me know if at any point you can't see my slides or something gets, you can't hear me. Just let me know.
Yeah.
So, yeah, you know, if you have ever seen fireflies or maybe even heard about fireflies, you probably realize that all around the world, these tiny insects create scenes like this one. This picture was a long exposure that's taken in the Smoky Mountains of the United States, or this one in central Japan. And these luminous landscapes fill us with wonder, even as supposedly objective scientists. And indeed, no matter what we call them, minmini or fireflies, these are one of the best ambassadors that we have for nature's magic. And it's because of this that for centuries, these insects have been celebrated in poetry and in art and in stamps all around the world. So in addition to their enduring cross-cultural appeal,
Madam, sorry for interruption.
Yeah.
Can you put full screen? Your slides are not showing full screen. Yeah. Yes.
Is this better?
Yes, it's better.
Okay. So fireflies also have economic value. So as I'll describe later in this talk, people go to visit sites all over the world to see fireflies. And this provides their economic value. It provides revenue for state and local governments, as well as livelihoods for local communities. In addition to their aesthetic value and their economic value, Fireflies also have great scientific value. So during the 20th century, firefly luciferase, which is their light-producing enzyme, was extracted directly from fireflies, and it was used for a variety of applications. Firefly luciferase was used to test for harmful bacteria in food and drink. Luciferase taken from fireflies was also sent into space to see if there was life on Mars. And luciferase from fireflies was used to quickly measure bacterial growth in biomedicine. More recently, the little bit of DNA that encodes the luciferase enzyme has been used as a reporter gene. So when this firefly luciferase gene is inserted into cells, it can be made to light up whenever a particular gene of interest gets activated. So as a reporter gene, luciferase has many applications, including finding genes that make crops more resistant to drought and testing new drugs. So fireflies have given us many gifts. I'd like to give you a roadmap of my talk about firefly conservation. And in order to talk about conservation, I really need to start with a brief review of firefly life cycles and a little bit about firefly reproductive behavior. I'll then talk about some of the threats that they're facing, both globally and more locally. And I'll describe some of the conservation work that's currently going on around the world. So you have already heard today that fireflies belong to the beetle family, Lamparidae. And around the world, there are some 2,200, probably more, different firefly species. And these come in a lot of different sizes, shapes, and lifestyles. Like all beetles, fireflies undergo complete metamorphosis, meaning that they go through distinct, ecologically distinct and morphologically distinct life stages. So they start out as eggs, which are generally laid in soil or moss, someplace moist, which hatch out into larvae. They go through several larval instars. The larval stage in most fireflies lives on or underground. After several larval instars, they will eventually transform into an immobile pupa, and then finally into an adult firefly. You may already have heard that most adult fireflies don't eat anything at all. All of the predation happens during the larval stage. So adult fireflies are typically rather short-lived. So as you can see here in this graphic, the adult stage of fireflies takes up a relatively small portion of their entire life cycle. Adults might live only a few weeks, but fireflies can spend months to years in one of their juvenile stages, particularly during the larval stage. These guys, firefly larvae, are voracious predators. And depending on the species, they might feed on slugs, on snails, on worms, on anything soft-bodied. And they are equipped with these extremely sharp sickle-shaped jaws, which they use to first inject paralyzing neurotoxins into their prey, which they then liquefy and ingest. Most firefly larvae are generally quite small, but as you can see in this group of Lampigera larvae, they can get to be quite large. So to complete their life cycle then, fireflies need habitats that can sustain all of these different life stages and their prey. Okay, so now it's time to turn our attention to romance. Fireflies have evolved many different ways to find and attract their mates. So there are flashing fireflies, where both sexes give quick, bright flashes to attract mates. There are also glowworm fireflies, whose females are wingless, and these females give long-lasting glows that attract flying males. And finally, there are daytime, day-flying dark fireflies whose adults don't light up at all. Instead, their females emit chemical signals, pheromones, that males detect using their rather more elaborate antennae. So in many flashing fireflies, courtship involves an exchange of flash signals between the males and the females. In some North American fireflies have evolved rather elaborate species-specific courtship codes, like the different species of Photinus fireflies that are shown here. And these different flash patterns have been shown to be used for both species and for sex recognition. And here's another remarkable evolutionary feat. This is a very rare, highly specialized courtship behavior that's known as male flash synchrony. And this is seen in just a few firefly species. It's known from several pteroptic species in Southeast Asia. You'll be hearing more about those shortly. And in these species, hundreds, sometimes even thousands of males will gather together in visually prominent display trees, like this one that's shown here, where they flash on and off in unison to attract flying females. Synchrony is also known in just a few North American species. In these cases, all of these species, it's flying males that synchronize their flashes with their nearest neighbors, producing what looks like a flash wave that moves through the forest. But earlier this year, I spoke with Sriram Murali, who described to me the synchronous fireflies that he has filmed at the Anaimalai Tiger Reserve in Western Ghats. And so, as we all recognize, India is home to astounding biodiversity. It hosts nearly 8% of global biodiversity on just 2.3% of global land area. So I'm excited that perhaps one or even several Abscondita species and perhaps other species from India might soon be added to this list of species that show this unique courtship behavior. Okay, so I'd now like to turn to a different topic, which is the fact that in many parts of the world, firefly populations are fading out. To better understand the threats facing fireflies in different geographic regions, in 2020, we, when I say we, I mean the IUCN Firefly Group, we did a survey of firefly experts around the world, asking them to score a dozen or so potential threats on a scale from 1 to 5. The results are shown here. These are the average scores, standard errors, for the top 4 threats: habitat loss, light pollution, pesticide use, and water pollution. And you can see that these top 4 are pretty consistent across all geographic regions, not everywhere, but in many places. Habitat loss and light pollution are perceived to be the biggest threats to firefly populations in that region. So let's take a closer look at these 2 top threats. So worldwide, the biggest loss of biodiversity in general, the biggest threat to biodiversity in general, is the loss of suitable habitat, in this case for adult fireflies and larval fireflies and their larval prey. Here are just 2 examples. In Malaysia, many mangrove-associated fireflies are threatened by the loss of mangrove forest, which is often converted to agricultural use, for example, oil palm plantations and shrimp aquaculture farms. In the United States, many fireflies are restricted to freshwater coastal wetlands. And here's one firefly. It's called the Bethany Beach firefly. And a few decades ago, its habitat along the coast of the US in the mid-Atlantic states looked like this. But now, more recently, it looks like this. This kind of coastal development and filling of wetlands has earned this particular US firefly the sad distinction of being the most critically endangered firefly on the IUCN Red List right now. Okay, so worldwide, light pollution is considered to be the second biggest threat to fireflies. This is a map of light pollution, also commonly known as artificial light at night, or ALAN, and this is based on 2021 satellite data. So we all know that over the past few decades, there's been an exponential increase in light pollution pretty much everywhere on Earth, leaving fewer and fewer dark places for nocturnal species to flourish. And light pollution is a problem for all nocturnal animals. It's a problem for humans, but it's a particularly big threat to fireflies because remember, they evolved to detect their bioluminescent courtship signals against a dark background. And in 2 recent reviews, we summarized lots and lots of observational and experimental evidence from many different firefly species that's been gathered recently, showing that artificial light disrupts courtship of fireflies. So I know everyone likes data, so I want to show you just a little tiny bit of experimental evidence. Just one example of one such study, which was conducted by my former PhD student, Avalon Owens, and this was published last year in Insect Conservation and Diversity. So in this study, Avalon went out into the field and she collected males and females of this particular species of firefly, and she brought them into the lab where she exposed them to 5 different types of LED light that was either bright or dim. And she video recorded the courtship flashes of both sexes. The title of her paper actually kind of gives away the take-home message, but I'll show you the data that what she discovered. So starting first with just the males. So this is male flash rate on the y-axis, and on the x-axis is different light treatments from complete darkness to cool white LEDs, warm white LEDs, blue, amber, and red LEDs, 5 different colors of light. And in darkness, she found that males flashed about 10 times per minute. However, when the males were exposed to any color of artificial light, their flash rates decreased to about 50% of what it had been in darkness. And this inhibition happened even when the lights were dimmed substantially, indicated by the striped bars here. Okay, so let's look at what happened to the female responses to these male courtship signals. So this is the female response rate as a percentage of the times that the male flashed that the female responded. This is the courtship dialogue, typical of this species. So in darkness, females responded nearly 100% of the time to any male courtship flash. These females are very receptive. They are ready to mate. But this female response fell to nearly 0% under all colors of artificial light. This is the bright treatment. This is the dim treatment. The dim lights were a little bit better, but please notice that the response rates of females are still significantly lower in any artificial light, dim or bright, than compared to what they are in darkness. Okay, so from this study and several others on North American and also European firefly species, we now know that light pollution disrupts firefly courtship. It reduces their reproductive success. But here's the good news. We already know enough about firefly behavior and ecology and their threats to start conservation efforts. And starting just a few years ago, we now have 2 international organizations of scientists and conservationists who are working together on firefly conservation around the world. So, using our existing knowledge, we're now identifying which firefly species are most at risk according to the standard IUCN Red List criteria, the gold standard for conservation of biodiversity. And we started last year with Red List assessments for US and Canadian fireflies. Right now, we're working on Red List assessments for Europe and Southeast Asia. And by sometime next year, we're hopeful that we will have Red List assessments done for Mexico, for Israel, and also for Brazil. And of course, if anyone is interested, we would love to help with Red List assessments of fireflies in India. Recently, our IUCN specialist group has been working with a North American NGO partner called the Xerces Society for Invertebrate Conservation. And in 2020, we published conservation guidelines and firefly-friendly lighting recommendations. And both of these documents are available freely downloadable as PDFs from the Xerces website. I've put the link here, and I can also put it in the chat later if you remind me. Okay, so I'd like to wrap up by talking about firefly tourism, because this represents a very, very important local threat to fireflies in many regions. Keeping in mind now the full firefly life cycle, it's clear that poorly managed tourism can have a big impact on local firefly populations in many ways, including the loss and degradation of both the adult habitat and also the larval habitat. And trampling from excessive foot traffic can harm any of the terrestrial stages, the eggs, the egg-laying females, the larvae and their prey, the pupae. Tourism also introduces more light pollution, which we already know disrupts firefly courtship. So in 2021, the Firefly group decided to take a detailed look at firefly tourism around the world. And we published this paper in Conservation Science and Practice. I'll mention just a few highlights of what turned out to us, for us, it was a very eye-opening review. When we tallied up the number of visitors country by country, we discovered that firefly watching attracts over 1 million visitors to sites in more than 12 countries. Some of the biggest are shown here. This is great because it creates economic opportunity at the national, at the state, and at local levels. And it's also great because having a firefly experience inspires a greater interest in biodiversity conservation on the part of tourists. So we recognize that every firefly tourism site has a unique set of biological, cultural, and economic factors, but we still came up with some general guidelines that we suggest might help promote more responsible tourism, including the need to protect habitat that's needed for all firefly life stages to thrive, including things like this raised walkway and a raised observation platform that's built over firefly habitat in Thailand, and a fenced off area, a walkway that's fenced off to keep tourists from walking through firefly habitat. We also suggest the importance of providing training for guides, as is done in Taiwan, on firefly ecology, life cycles, and behavior, and educating the public about these things also. We also suggest the importance of encouraging local communities to participate as key stakeholders in designing and managing tourist operations, like the Firefly Guide community that the Malaysia Nature Society has set up in that country. So working again with our North American partner, last year our IUCN group put out several publications aimed at making US firefly tourism more responsible. So including this guidelines for site managers and visitor guides to improve what we call firefly watching etiquette, so tourist behavior at firefly sites. And again, these are all freely downloadable PDFs available on the Xerces website. So with that, I would like to thank the Fireflies, and I would like to thank you, and I would be glad to try to answer any questions that you might have.
So thank you very much, Professor Sarah Lewis, from the life cycle to reproductive behavior, threats, ecotourism, and conservation. So we had a very broad view of the initiatives which the IUCN has taken up in some of the countries. Now, one basic question is that how to start work on conserving fireflies. Now, especially we are working in Western Ghats, and you have already shown one slide of Annamalai forests. And we have similar evergreen tropical forests in this part of the South India. And now in October, we would like to make some initiative, make people aware of what is the importance of this site, what is the importance of fireflies, and how we can conserve those sites. I would like to know that how we can rope in IUCN and your kind self in this kind of efforts.
We're happy to help in any way we can. So I think one of the important things, and perhaps you already have done this, is to know a little bit more about the behavior, about the life cycle, identify the species of firefly, and that will enable us to get some idea. Are we looking at several? Are we looking at one? Are we looking at a dozen different species? And maybe get some information about their geographic distribution within India. So that would be, that's, I know it sounds like a lot, but kind of a good place to start in terms of IUCN red listing. Of course, in terms of conserving fireflies, it's not always necessary to know everything about them. And so if these are fireflies that have, for example, a terrestrial land-dwelling larval stage, we already know that there are certain things that we can do to protect the juvenile stages of those fireflies. And there's a wealth of information from so many other countries like Taiwan and Thailand and Malaysia who have successfully, really been incredibly successful at, well, not always like right away, but have learned maybe the hard way how to protect the fireflies and still host very, very large tourist inputs of tourists to these sites. So I think that's a great thing. There's so much to learn from those places, and I'm sure that you have connections. So yeah, and again, for if there's anything that we can do, we're really, the IUCN group is really eager to help in any way that we can. Happy to share information, happy to help map distributions, happy to write red list assessments. If you have the experts there, we can help with some of the sort of logistical stuff. So yes. I hope that's helpful.
Yes, surely. Only thing is that we'll have to work together and see what sort of help and also the legal help. Now, there's one question from Miss Aditi Rao. She says that in Western Ghats, most of the natural areas are being converted to cultivation. This is out of the economic compulsions. They have economic compulsions. So how do we stop these kind of transformations, and how do we stop also the application of pesticides?
Yeah. So great question. Thank you so much. I guess there's two things I'd like to say. One is that, habitat loss is the greatest threat to biodiversity in general. So if there's any way to prioritize existing natural areas for conservation, obviously, we need to feed people. Agricultural conversion is going to happen no matter what. So I guess one thing is sort of prioritizing in terms of biodiversity areas and including fireflies. That's a tricky question in every country around the world. Getting the powers that be to think about these tiny insects and even larger, larger, let's say, equally charismatic megafauna. So this is indeed a very tricky problem for conservationists that we've dealt with for a long time, many people much longer than I have. So I don't have very good advice on stopping agricultural conversion. In terms of pesticides, I think there's more and more recognition. Last year, I think, or a couple of years ago, the European Union managed to ban the outdoor use of neonicotinoids on agricultural land, and that was a remarkable feat. These pesticides are still widely used in many countries, including in the United States. And we know that these insecticides have harmful effects on many beneficial insects, non-target insects. And so having some way of regulating, especially in surrounding natural areas that might eventually be protected, regulating pesticide use locally or would be is a really, really important thing to work towards. I also wanted to say that protected areas like the Tiger Reserve are fantastic. And one of the things, however, that we are realizing is that even these protected areas are permeable to light pollution. So in places like Brazil, where there are, again, like India, many, like a mega diverse country, many firefly species, and many of those species are in protected areas. It turns out that they're still vulnerable to light pollution because that just seeps in through the boundaries of the protected area. So that's something that especially firefly conservationists are beginning to think about, is how to provide protection in protected areas from this ubiquitous threat to fireflies, to flashing fireflies. So a question in the chat from Arpan. Asking about areas of research where young scientists can contribute to information that's needed for conservation. Really, thank you so much for that question. It's a really, really important that we, we can't do anything without young people getting involved. And there's 2,000 plus species of fireflies, and there's so much we don't know about them. And as Dr. Ballantyne has certainly said, there's a lot that we need to learn about the identity of the fireflies in India, the life cycle, their behavior. And I'm really, really encouraged. Actually, last month, many of us were at a meeting in Portugal, the International Firefly Symposium, which is typically held every 3 years, was held in Portugal. And one of the things that I was like so delighted to see was how many young people are working on fireflies and working on firefly conservation. For example, in Mexico, in Brazil, in Malaysia, in different European countries, many young people are looking at their local firefly species and looking at the effects of light pollution on that particular species. And it's interesting because it turns out that for I showed you data for one North American species, but there's other species that fly earlier in the evening, like just after sunset. And those species seem to be less susceptible to their courtship signals being disrupted by artificial light. So looking at, so we know there's going to be differences among species in their visual ecology, what they see, what lights they react to. And this is a really important area for young researchers to get involved in. So thank you for that question.
Yes. Another interesting question would be, and it's really practical question, that does ecotourism and conservation go hand in hand?
Yeah. I'm sure that many people here know more about this than I do, but obviously, you know, poor ecotourism is just a label. And I think that just as there is poorly managed tourism, there's also poorly managed ecotourism. So it's easy to put a stamp of eco on this behavior of, you know, bringing people out into the natural world to see some of these amazing creatures of all kinds. And but there also this needs to be carefully thought about and carefully designed. And so the paper that that I mentioned, is just like a first step in trying to make some suggestions. And again, like every place is unique, and every tourist site, potential tourist site, has different firefly species, different ecology, different cultural and political, biological factors. And so it's just sort of something we're hoping that people will keep these things in mind as firefly tourism is developed in other countries. And again, you know, we're happy to work with anybody in trying to look at local factors and to you know, provide advice as much as we can. We're not that knowledgeable, but we know a few things.
Yeah. Yes. Professor Sarah Lewis, you have quoted that there have been successful examples of conservation, maybe at local level in Thailand, then in Malaysia, in Taiwan, and other countries. But what I see is that in most of these developing countries, there is no legal protection accorded to fireflies as yet. For instance, in India, we know very well, and these fireflies, although not scientifically connected, popularly they are connected with the local people. And why not IUCN make an initiative to these governments of these countries to protect fireflies vis-a-vis habitat and the mega species?
Sure. Yeah, you know, if you think that would be effective, we would love to be able to write letters, to send emails, to provide information to any experts and any politicians at the local level who you think might be able to help conserve some of these natural areas and to protect the fireflies that live there. And so, yeah, the only countries that I know of, you're right, there is no legal protection for fireflies in any parts of the US. There's no legal protection for fireflies in Europe. The only countries I know of that have legal protection for fireflies are a couple of species are protected in Taiwan, I believe just 2, and in Japan, a couple of species and their habitat, still known as Luciola cruciata, I believe, although Leslie, correct me if it's changing, but the Genchi firefly in Japan, its habitat is protected. And so it's very, very rare. And it does, as you say, you know, it comes with a, in the United States, there's sort of a weird, I'm going to say that the United States is not high on the list of countries where people have a cultural appreciation for fireflies. And, but in countries where there is inherent, longstanding, you know, thousands of years appreciation for fireflies, as in many parts of Asia and Southeast Asia, and I think that maybe legal protection for fireflies is, there's more political will to do something like this. And yeah, again, we are absolutely eager to help in any way that we can. I'm happy to talk more about it. I'm so sorry to have gone over my time. I really apologize.
Yes, there's one question from Ranjini Swaminathan. She says, why the male flashes are getting affected by the artificial light exposure than the females?
Yeah, that's a really great question. So so we know that it's happening, but we don't know why it's happening. So it could be that that artificial light, for example, makes males, it like disorients them in terms of what time of day it is. So they're cued by dropping light levels as it gets darker. Different firefly species have a particular level of light that they will begin to flash at. So on a cloudy day or a cloudy evening, fireflies will flash earlier than if the skies are clear. And so it could be that that light pollution inhibits their detection of what time of day it is. And they just think, oh, daytime, just gonna hang out. It could also be that it inhibits, instead of like temporally disorienting them, it might just somehow inhibit the pathway, the neurological pathway that results in flashing. So the short answer is, we don't know. Great question. You should study that.
Okay, that's a good, opportunity for youngsters to explore. Yeah. Then this is just a personal question, Professor Lewis. See, we find that there is Southeast Asian countries, especially the Indian subcontinent, are a high biodiversity spot, maybe for fireflies too. Would you think that will it be a good idea to establish a network of all the firefly workers in these Southeast Asian countries, which can act adjunct to the IUCN group?
Sure, absolutely, definitely. Yep.
Yes.
Yeah, I think that's a great idea. I also want to just say that, you know, the IUCN group, we're a pretty small group. There's like, I think we have like 30, 40 members, and we are actively looking for more members to join our group from other countries. So please, yes, however we can help, glad to.
Yes, that's true. Any other questions? There's one question from Nishant Venugopal. Generally, people are not aware of the pest control benefits of the larvae of fireflies, because they are carnivorous, and sometimes and most of the times they're feeding on the snails and slugs, which at such situations can be pests. So any reading material on this?
Okay, that's a really good point. So, again, with 2,000 plus different species, they have different larval feeding habits. So not all fireflies feed on snails, not all fireflies feed on slugs, not all fireflies feed on earthworms. Some of them are very, very specific to like one particular species of snail. And I think you may hear more about that shortly. Some of them have a very broad diet. And as it's been pointed out, like very few people have raised larvae and know what they're eating. More importantly, I guess, know what they're eating in the field. So sometimes you can raise larvae on different kinds of prey than they actually are eating in the field. So there is a paper that appeared last year, and I'm not going to remember where it appeared, but it's a scientific paper that was basically a review of larval biology of all Lamperidae, and I highly recommend Googling that. It's by Luis Silveira, who is a Brazilian researcher who's currently in the United States, and I think he's the first author on the paper. And it's excellent. It has a review of larval biology. So that basically has everything that we know, like a scientific paper. So maybe not like something you want to distribute to everybody, but it could be a good source of information. I hope that helps.
Yeah. Mr. Parvez has one question. Yeah. Parvez.
Good evening, ma'am. Thank you so much for your wonderful lecture. And I just wanted to know, like, what are the opportunity IUCN provide for the young researchers doing like firefly research, like globally?
So say that again, please.
What are the opportunities can IUCN provide like for the young researchers? Like in the funding.
What are the opportunities?
Yeah. Thank you. I'm sorry. I'm thinking that great question. I guess, networking is one thing. We have the IUCN has a incredible network of conservationists worldwide who work on invertebrates as well as other conservation of other biodiversity. So I think there's a growing sort of group of people within the IUCN who are employed by the IUCN who are eager to help scientists and specialist groups to compile information towards conservation of fireflies around the world. And we see that we just actually started doing the red listing of European fireflies and Southeast Asian fireflies. So for example, if you wanted to have the IUCN would be willing to host a workshop or to bring people together or to a workshop on conservation of fireflies in South Asia. That's something that the IUCN could certainly, we could submit a proposal, or you could submit a proposal to fund a workshop like that. So there are sort of a networking and a lot of expertise in conservation. As an ecologist, I have to say, like, I wasn't always interested in conservation, but I've been really, really impressed in the past 5 years since I've been co-chairing the IUCN group, really impressed with the vast amount of knowledge, and many of you already know this, that exists in the IUCN network, and they're really, really willing to help. So expertise, software, knowledge, facilitating meetings, providing information, all kinds of things. So yeah, thank you for that question.
Thank you, ma'am.
Yes. In India, we have butterfly, dragonfly festivals. And we are also connected with the citizen science initiatives. Hope there will be some initiative like that on fireflies in India. Any suggestions?
Yeah, that's a very, very powerful tool. And in fact, it's been interesting because as we've been doing the red list assessments for Europe, there are many longstanding citizen science platforms in different countries, in different languages in Europe for citizen science. And so many of the observations, so uploading photographs, obviously, many of the observations have been verified by Firefly researchers working in Europe. And so we've greatly increased our knowledge of the distribution of many species in Europe based on citizen science platforms. And that's a really, really new development. So I guess that the only advice I have is to make sure, like, so for example, there's a platform in Spain, there's one in Belgium, there's one in France, and each of these is sort of really kind of curated, let us say, by a Firefly expert or two, who, when people upload their observations, information on the flash pattern is really important. Photographs, Leslie will know more about this, photographs of the dorsal side, the ventral side of the beetle, very important. Maybe a close-up of other parts of the anatomy would be very helpful. And so, yeah, maybe having some guidelines for the citizen scientist to follow when they upload their observations, a little bit about the time of night that the fireflies are flashing. Obviously, they're geotagged, so the location is going to be there automatically. So the more information that can be collected, the better able experts will be to identify the species of firefly and then be able to look at geographic distribution of the different species. Is that helpful? Let me just, sorry, oh, you're muted. Still muted.
Am I audible?
Yes, now you are audible.
Yes. So we handsomely thank you, Professor Sarah Lois, for this effort and also for spending us so much of time and sharing with us your worldwide experience on the fireflies. The information has been very informative. It has been educative and helpful. And surely these suggestions will be taken forward and put into action. So that's what we are interested in. So we would be continually in touch with you, and we look forward to have many more such inputs in the times to come. So thank you once again, and we hope that this webinar will definitely contribute to firefly conservation and education. Yeah. So now we move on to the last speaker. Is Dr. Nada Badruddin here?
Hello. Hi.
Yeah, you're here. Yeah, so you could complete your touring and your survey, and we are very happy that you could make this what you call a webinar by your gracious presence. Now, to introduce Dr. Nada Badruddin, she has been with the Forest Research Institute, Malaysia, and she has been particularly working on congregating fireflies. Now, this is one very important aspect of firefly conservation, because many people witness thousands, lakhs, and millions of fireflies, adults flashing the light at the dark nights. But we are not sure as to how we can monitor and conserve these congregating firefly populations. And we are glad to say, no, that Dr. Nada has developed a non-destructive method to monitor these congregating firefly populations in any area. And this has been extensively used in Malaysia. And later, she expanded her research scope to include the fireflies in biodiversity of tropical inland forests, and also the ecological influences moderating their distribution. The Firefly group is relatively understudied compared to the congregating fireflies in Malaysia, and how species composition varied with habitat characters have been better understood through her studies in Malaysia. She's also a part of the Firefly Specialist Group of the IUCN SSC, which is currently assessing the conservation status of the congregating fireflies of Southeast Asia. Dr. Nada, you're most welcome to this webinar, and please, you can begin your lecture. No, she's still unmuted. Yeah, she's yes.
Okay, hello, thank you.
Yeah, thank you.
Yeah, just now it was, I cannot mind. Okay, so I'll be sharing my screen now.
Yeah.
Before we start the, okay. Are you seeing my screen now?
Yes.
Okay.
We can.
All right, let me see what should, okay. Thank you. Thank you, MPRI India, in particular Dr. Chakrabarti, for having me today in your first ever Firefly webinar in India. I am happy to be around everybody here. To Dr. Leslie and Dr. Sarah Lewis is also around to listen together on my talk. So essentially, for today, I would be sharing with you what are our experience in conducting Firefly research, in particularly for Malaysia, and focusing on FRAME's efforts. And it is hoped that at the end of my sharing, it would give some idea to you all, especially in India, the researchers and the academicians, on how to approach Firefly research, and if there's a set of target, if it's on conservation, what are the steps that maybe you can follow through to make sure that the efforts are more smooth, and you are able to capture information that is beneficial and useful when it comes to conserving the fireflies. So, sorry. So for today, I'll be sharing on the Malaysian fireflies. I'll be talking a bit on briefly about the types of fireflies we see in Malaysia and what are the threats that they face, and going through also our experience conducting research, what aspect was covered, and what were the findings, and eventually how the findings help us not only to increase our understanding, get the basic information of fireflies, but also to assist in planning and managing proper conservation approaches. So in general, when we talk about the fireflies in Malaysia, we can group them into two groups, which is the solitary fireflies and the congregating fireflies. So these two groups are actually quite distinct between each other, whereby the solitary fireflies, they occupy numerous habitat types. They can live in lowland areas, inland forest areas of lowland dipterocarps, but we would be seeing them up to the mountaintops as well. And when we are comparing their species diversity with the congregating fireflies, they are actually relatively higher in species diversity if we are to, for example, sample in a location in one night. We will be able to collect up to 7 or 9 species of fireflies. But if we are going to the congregating firefly habitat, they are usually in low number when it comes to the species richness. However, when you are talking about abundance, they are lower in a number within a certain location. And their way of flashing their light is mainly while on flight. For the congregating fireflies, for Malaysia, we are actually blessed to have these congregating fireflies because globally, they are not really widespread. They are more towards our Southeast Asia region. I'm informed from records that I've read, they also occur in parts of India, but that has to be confirmed by the Indian researcher maybe. And these congregating fireflies occur in Malaysia, and these are the fireflies that are currently a tourist attraction, like what Prof. Sarah Lewis mentioned just now, attractive to the public to see them while riding boats. So in comparison, these are the two differences between the solitary fireflies and the congregating fireflies. They are confined to river estuaries where high tide and low tide are being experienced by the river. So they receive salt water from the sea and also fresh water from upstream. And when we talk about their abundance, the adults, we would be seeing them in large groups displaying their lights on trees that line the riverbank. So around the world, like what Prof. Lewis mentioned just now, all fireflies are generally under threat mainly by human modifications to the habitat, pollution, and also we are seeing the climate change impact also to the fireflies. And because of the fireflies' confined distribution, some fireflies, they have flightless females, whereby they cannot move much from where they are found. And also, like for example, the congregating fireflies, their habitat is very specific to the river estuaries. So any changes to their habitats means that they are very susceptible to a negative influence. Okay, so these are the threats that we identify in particular for Malaysia, but I think it applies to all as well when it comes to the firefly and how it impacts the fireflies. And 3, they can be grouped by the habitat loss or change, pollution, and climate change. And I'm elaborating a bit about the habitat lost from land development is usually for agriculture, for urbanization. And when it comes to flood mitigation, although it's important for humans, but some flood mitigation strategies are impacting the fireflies, whereby, as for example, for the congregating fireflies, where the rivers were widened to take more water when there's excess water from upstream. There's also flood mitigations from doing bunds along the river, which disturbs the breeding habitat of the congregating fireflies. There's also water extraction from the river, which can reduce the river flow and also influence the salinity of the river water as well, which impacts the water quality. And we are also seeing impacts of logging towards the inland forest fireflies, which is the solitary fireflies, and also the other pollution and climate change, which Prof. Sarah also did mention about light pollution in detail before this. Okay, so for FRIM, we started doing firefly research in the early 2000s. And at that time, the firefly tourism, in particularly for the Selangor River, which is situated in a coastal town of the most developed state in Malaysia, has become more popular as a tourist attraction. But at the same time, realizing the area, being in a state that is most developed in Malaysia, is not spared from development. And therefore, we need to understand the fireflies there first. And to do so, we started at the basic information, which is to understand the life cycle. So in order to do so, we did a small scale captive breeding with the main objective to understand the overall life cycle, which is important in piece of information before we want to understand how to conserve a certain species, for example. Because different species of fireflies require different approaches. We cannot have a one standardized way of conserving. It can be generalized, but there are specific approaches that has to be applied. Therefore, we need to understand the life cycle, what they require, the biological requirements, the ecological requirements before we can further expand our knowledge and understanding of the firefly. So from this study, not only we managed to understand the life cycle, but we also understand where they occur in the habitat. We got to understand that when we see the fireflies on the trees, it's just them as an adult. But the tree life stages actually occur at the back of the display trees, the habitat for the display of the adults. So it's critical to look at conserving the fireflies, not only the adults that we see, but to safeguard also the three life stages that they are in order for them to complete their life cycle. So when we're talking about the congregating fireflies, we know that they need these two habitats, the breeding habitats and the adult habitat, but 80% of their life is actually spent in the breeding habitat. The three life stages actually happen in the breeding habitat. And yet, at the same time, it's these areas that are being converted for development into, for example, plantation or vegetable farms, or even for residential area. So, because we understand, at least now we understand where are the important areas for the fireflies, not just the display trees, but also the areas at the back of the display trees. So we then later proceed to compare the different landscapes within the habitat of the congregating fireflies, and compare how the firefly larvae abundance occurrence in these three types of landscape within the firefly habitat. And from the study, it was found that they are more successful in the natural area. They didn't do so well in human modified areas such as the orchards and the plantation, mainly because of the changes to the microhabitat, where the grounds are much drier. There's no occasion of inundation from the river because the people build bunds to prevent flooding into the area where they plant their trees and plants. So, and there's also a very obvious fluctuation of temperature in comparison to the natural area, which has a more stable microclimate. So, all this information we are continuing to capture from our research. And then we later embark on firefly population monitoring, which Dr. Chakravarti mentioned a bit in the introduction just now, where it's not actually me that developed the method, it was actually a work of a team. and I was one of them that were involved in the work. But the question that was raised is that by the time we reached this point, the caretakers of the river, our the water management agency, now has realized the importance of taking care of the river and the fireflies, and they ask, okay, so how can we know whether the fireflies are actually getting better or is it worse? How do we decide? So that's one of the reason why we initiated this population monitoring program. We wanted to develop a method that can be applied and utilized in the long term. Therefore, we have to identify a way that we can monitor without disturbing the fireflies in their area. So this firefly population monitoring utilizes night photography of the fireflies. And what we do, actually currently we are still collecting data of the firefly population, is by setting up a camera across the river from where we are monitoring the stretch of display trees of the adults, and every month our team will go in the area in the same permanent monitoring sites along the river, which captures the range of habitat of the congregating fireflies in Selangor River. And we take images of the fireflies on the trees and program for the computer to calculate the numbers of dots of the light flash, and from there we come up with the index of firefly population abundance. So what we found when we compile all this information is that yes, the firefly is declining, and we continue to collect data of the population, because in between these years there are efforts of helping the fireflies. So monitoring is important to be formed to assess whether we are doing the right thing when we are implementing some conservation approach or strategy. Is it effective? So monitoring is a way to assess this, and so therefore we continue to do this monitoring. And at the moment our monitoring is only in Selangor River, in Selangor, but this method has been it's not applied, but I think it was modified by US to also implement this technique of photographing the fireflies for monitoring their population. Okay, so besides getting the trends of the firefly population, because we are going into the site every month to photograph the firefly trees, the firefly displaying trees, we are also able to detect changes to the habitat. So this is 2 sites that were monitored, and that, as you can see here, the lighter green canopy are the display trees of the fireflies. And at the back there, which comprises here more of the nipa trees, are actually the area where the firefly larva, egg, and pupa lives in. And from the photograph that we took, we were able to detect the land clearing that happened. And with the information we have, we went to the site and we saw that there's some land clearing happening in the area behind the brembang tree, the display trees. So the display trees are intact, as you can see in the previous slide. When there's changes, the display tree will not harm at all because it's right at the river edge. But the clearing happened at the back of the display trees, which actually disturbs the fireflies. And this is reflected by the, if you can see here, that happened in 2009. And it was supported by a sudden decline of population in Selangor River at that time. But because of the information we have, because of the record that we have, the images, we were able to alert the authorities of the things happening. And in 2009, the area were gazetted as a firefly protection zone. And it means that any development in the area is more controlled, and therefore with the hope that it will minimize any development impacts towards the firefly population in the area. And then we proceed on to understand the inland forest fireflies, because all this while, for the past 15 years, our focus had been on the congregating fireflies. We have not barely touched anything to understand the solitary fireflies, which, as you see, has a higher diversity level, meaning that they depend on a much complex habitat for them to sustain themselves in, and having different diversity. The higher diversity means their requirements are different and very complex and varied. So the study was to understand how logging impacts the firefly population, because when there's logging, we would be seeing canopy gaps become larger, therefore increasing light penetration to the ground. It will also impact the soil temperature where the firefly larvae occur, and where the snails, the prey of the larvae, occur. And also because of the less trees, there would be a decrease in leaf litter, where fireflies are known to be in, the larvae will be resting under there. The female fireflies would be also depend on this area to keep themselves at an optimum condition, and also with a more open area, there would also be an increase in the growth of understory plants. And how this impact the fireflies is that it would increase the light penetration, even night light, and you can increase the soil temperature, which affects the firefly larvae and the prey and their prey, and so on and so forth. And from the study, we compared between areas that were logged twice and only logged once, and we found that species richness of fireflies in once logged forests are higher, that were recovering from once logged activity are much higher than area that has been logged twice, which means that the recovery is not enough to bring back the number of species of fireflies that we were seeing in the area before it was logged. So probably some revision of the way the logging management has to be considered as well. And then we also understand the climate change impact that could happen on the fireflies. So we also conducted studies across elevation to find out more information about the occurrence of fireflies across altitudes, and not only to understand how climate change may affect the fireflies, but it's actually a way to do a rapid assessment of the fireflies, because as I mentioned, when it comes to the solitary fireflies in Malaysia, there are barely no information about the solitary fireflies. So we want to get as much information as quickly as possible, and studying across elevation is a way for us to cover different habitats at a shorter period, Therefore, we embarked on this study, and from that study, it was found that fireflies are distributed at certain elevational ranges, which means that they may not be able to survive, especially for those in high altitude location. If climate change increase temperature, then the condition will not be suitable for them anymore. They might not be able to survive further. And also from the study, it was found that when we're comparing from the mountains that we surveyed, they are the beta diversity is more attributed to the species replacement rather than species loss, which means certain species only occur in certain parts of the forest that occur along the elevation. So we won't be finding the same species in certain all the sections of the forest. So that's something that is important to bear in mind as well. Meaning, if we want to we want to maximize the conservation of fireflies, the numbers, the diversity of the fireflies, we have to take into account all the different habitats and not just focus on certain areas of the inland forest. And when it comes to this sampling of the fireflies, because for Malaysia, I think that the study that we did was one of the first to focus on the solitary fireflies. So we have designed a method for sampling that captures the information of the firefly, and why we conduct the collection at night and still collect the larva is that it also in general captures the information of not just the nocturnal fireflies but also the diurnal fireflies, because all firefly larvae are active at night, and therefore at least with one survey at night we still can capture information of both the diurnal and nocturnal fireflies. And the habitat features that were measured that we've postulate has an influence on the distribution of the fireflies are leaf litter depth, canopy closure, height of understory plants, presence of water bodies, and proximity to primary forest. And upon analysis of all these habitat features, it was found that species richness of fireflies are affected, and the four of these habitat features are affecting the fireflies quite substantially. The leaf litter depth is affecting their presence, the canopy closure, presence of water bodies, and also the proximity to primary forest. These are the four habitat features that impacts directly on the presence of the different fireflies in a certain areas. And indirectly from the study we did, with the help of Dr. Leslie Ballantyne, we were able to describe new species to science, and it's a good record of it has improved the record of fireflies in the country substantially, and it also shows that when we find new species, indirectly it shows that there are more to be explored and to be discovered. So I'm looking forward to that as well. I'm keeping in touch with Dr. Leslie Ballantyne. Even as we are speaking, I'm also still close contact with her to know and learn further on how maybe we are also seeing more new fireflies coming from Malaysia. And the most recent work that we have done completed, but there could be an expansion of this, is that with involvement of the public, the local community in with researchers, and this is a good, I would say, a good example for Malaysia as well, because so far that I know, this is an initiative by a local community organization that wants to keep their area a patch of a green area in their housing area, a green area for them to admire and appreciate nature. So they have engaged FRIM and also Urban Biodiversity Initiative to conduct a preliminary study to develop a citizen science protocol to assess the Bukit Kiara is the area, firefly community and habitat. So here I'm sharing some images of what we have done, the interaction we have with the citizen scientists and the volunteers, and how with their involvement, it actually help researchers to get more information if we compared to if we just rely on our own in getting data from the field. So there are several activities involved where we actually bring them to the site to train them, and there's also workshop, and also once we have developed the methods, which we go together with them, but eventually they are able to do it on their own. And we also have Firefly ambassadors. They call them the Firefly ambassadors, that are the main players in coordinating the citizen scientist program in Bukit Kiara. So some of the interesting findings from the project is such as Lampreygira female nest found in the ground. We were able to capture information of the firefly feeding behavior at larval stage, and also possible predation of the firefly larva. So there are so many interesting information that we get from the study, because we are able to do it together with the public. And these are the types of fireflies that were found in the area, the snails record, and also this is a Lampreygira, is actually the iconic firefly in the area. Yep. So in conclusion, when firefly conservation research actually needs to start from the basics. First, to have the species identified, where you need the taxonomies. And then we then later go towards the understanding the ecology, the biology. Then we need the biologists, the ecologists to come in. And once we have all this basic fundamental information, we have an idea what they require, what's important, where can we find them, how can we keep them sustained, survive. Then we can eventually do conservation status assessment, create awareness, engage the general public, stakeholders, and policymakers, because we have information to share with them, to advise them correctly. And FRIM is committed to continue firefly conservation work in Malaysia. And I hope this sharing of information, our experience, we are also still learning. There are so many things that we still don't understand about our fireflies, but I hope if not much, at least there's something that you can bring back and maybe modify and apply in your country, in India. So with that, I thank you.
I think Firefly workers in India and other countries have to learn a lot from your experience. And definitely we have been very much enriched on different aspects, on array of aspects on Fireflies. So you have touched upon solitary to congregating, from riverine habitats to mountainous habitats, okay, and from estimating the numbers to
How do I stop sharing now?
Huh? You're not
How do
sharing? She's not sharing?
How do I stop sharing?
Yeah, it's okay.
How do I stop sharing my screen?
Just a minute.
Ma'am, there is a icon in on the top. Stop share.
You can just icon on the top, right side.
Oh, stop. Oh, okay, stop sharing. Okay.
Yes.
Found it. Okay.
Yeah. Thank you very much. Indeed, we enjoyed and workers in other countries of Asia have to learn a lot about from your experience. Definitely we have been very much enriched and hope that some of the successful efforts will be contributing to our research, training, and outreach activities on fireflies in India. We have a lot of things in common also, so that will greatly help us from your experience. Dr. Neda, I would like to ask you that you have developed an index to estimate the numbers of congregating fireflies, isn't it? And then you have also used photography and digital photography to our camera traps to count, to get an estimate of the numbers in congregating populations. Now, do you think that we can also deploy the leader or drones or unmanned vehicles so that we can make a rapid estimate of the population, and with very less effort and maneuvers expended, I think there is a good scope to have devices deployed in drones, leaders, and other unmanned vehicles. What are your comments?
One thing about digital photography of the firefly light is that we need a very good set of camera with very high sensitive, what do you call the sensor?
Yeah.
It can be tried out. The method that we use for the congregating fireflies, as I mentioned, we only have so far do it in Selangor River. We are currently testing the same method for the other rivers, but because of the settings, the width of the river, all that have to play. So there's also some amount of research that needs to be done, and your approach of doing something that is not non-man, non-human way of maybe can, but it has to be tested and yeah.
Another important practical question is, Dr. Nida, see, the as far as the firefly ecotourism is concerned, you know, people who are not local, people who are not connected to firefly, or people who are not even connected to the firefly habitat or the forest will be benefited from firefly ecotourism. But the native or the local community will ask us a question: in what way this is going to benefit us? In what way the benefits of firefly ecotourism be shared among the local communities who are in fact the guardians of firefly populations?
Yes, a very good question, Dr. Chakrabarti, because we are also facing that. We can say it's like something like a conflict because even within the local community, at that whole stretch, not all are involved in the firefly tourism. But they do, what do you call it? They acknowledge that the presence of the fireflies in the area make the area more developed. They do, but at the same time, they need something that would directly have benefit to them. So what we are currently doing for the Selangor River firefly, because our experience with the congregating fireflies are mainly focused in the Kuala Selangor, is that we are proposing conservation action plan to the government, the state government, which includes giving incentives. For example, giving incentives to the local that, for example, maintain a portion of their land to not disturb. Means that even though they don't disturb the area, the patch of land, a bit of their patch of land, that where the firefly larva, the breeding area, they still can get money. It's usually it's dollars and cents. So we as a research institute, we can propose, but it all depends on the, as you can say, maybe the political will of the of the government. But if the governments themselves understand the value, not just value in, again, in the money sense of the thing, the value of it being something that you won't get to see fireflies like this in other areas. I suppose they would understand the need for involvement of locals in the community that is not directly having benefit from firefly tourism. So one of the things is giving incentive for them to retain their areas so that it's not developed. And also because in the area there are plantation, like the study that we did, because there are plantation, there's also, we can also engage with the agriculture department for them to do the best practice agriculture incentive as well. So things like that, certification. So they still can do plantation, but they get benefit from certifying their plantation. So which means they have to do certain criteria for conservation. So these are all the suggestions that can be applied.
Yeah, surely. And this has a wide applicability of all your suggestions, and definitely they have practical value. Now, another tantalizing question has been that the larvae are carnivorous. Now, in Malaysia, we know that we have sago palm, we have oil palm, and in India, we have coconut palm, we have areca palm, we have toddy palm. And here, the snails and slugs pose a threat because most of them are cultivated in humid conditions and under shade conditions where we have only filtered sunlight. Any effort has been made to macerate the larvae and see that the problem of snails and slugs, especially in palm cultivated ecosystems, be explored?
In our country, no. We don't see the, because fireflies in our area, if you are talking about the congregating fireflies, they are very small.
Yes.
And they feed on very small snails that are usually not the pest of agriculture crops.
I see.
And also if there's changes to the plantation, well, palm plantation are usually not affected by snails. They are more of from the beetles and others.
Beetles, defoliators,
beetles, moths.
True. Now, why I was talking about this is that we can definitely stimulate and encourage farmers to use this so that the it will have an added value to our conservation efforts, because you have been trying very hard, your team has been trying very hard to conserve and also prosper ecotourism. So if a section of people like the farmers could give a very sound advice to the natives that these are really beneficials, then definitely it will add in a major way to your conservation efforts. So that's one thing. So have you visited India anytime?
Never got the chance to be in
But
India.
you should explore. You should explore.
Yes, but
Definitely.
given the opportunity, I would love to come to
Yes,
India.
definitely. We would be very much happy to share your experience with us in the field. Okay, and also conserve the congregating populations. So, friends, any more questions from the participants, from the youngsters who are with us? Any others? Any questions to Dr. Nada Badruddin? Okay, then we thank you immensely, Dr. Nada, for sharing your experience, and definitely we will be in a major way benefited from your work.
Thank you.
Thank you so much, and we will be in touch with you. Okay. So please do visit India anytime, because we are almost not very far from your country and we face similar such situations. Yeah. So all these speakers from Dr. Leslie Ballantyne to Dr. Sarah Lewis, Dr. Chandana, then Dr. Barua, Dr. Gupta, then Dr. Nada, all are welcome to India. And we shall be there with you to sort of support you in your expeditions, in your explorations. And definitely we are always here to collaborate and cooperate. So thank you very much, Dr. Nada. With this, we end this webinar, and as a formality, just not the formality, but as a custom, we would like to thank all the speakers. So in the morning, we had Dr. Leslie Ballantyne, then we had Dr. Chandana, then we had Dr. Devanshi Gupta, then we had Dr. Anurup Barua, then we had Dr. Sarah Lewis, and then we had Dr. Nada Badruddin. So thank you so much for your valuable input and time. And we would also take this opportunity to thank our Director General, Dr. Jagmohan Sharma, the directors, the Director of Research, the coordinator of Malaysia office, the technical team of MPRI, the NGC team of MPRI, Madam Jennifer, then we also had the Naturalist School. So they all have contributed to the success of this webinar. The proceedings of this webinar will be circulated to all the participants of this webinar, and we also hope that the meeting, the International Firefly Conference, which was recently held in Portugal, the proceedings will be available, so that we will be very much benefited from the conclusions and from the recommendations the conference gave us. So I think this type of collaboration, cooperation, and coordination will go a long way in conserving fireflies. And we will have other initiatives also. We are going to have a walk. We are going to have a flyer for the common people. Then we are going to have a festival. So all this will be sort of intimated to you, and your inputs are always welcome. With this, we end the webinar, and thank you so much. Thank you all.
Thank you very much, Dr. Chakraborty, for all your efforts. So for all your efforts, it is a very
Yeah.
present experience. So
Yes.
many thanks.
Thank you. Yes.
Thank you so much.
Yes. Yes.
Great webinar. Thank you.
Yeah. Thank you. Thank you.
Thank you, sir,
Yes,
for
sir.
this webinar. Thank you very much,
Yeah.
sir.
Thank you so much, sir. Yes. Yeah. Thank you so much. Yes.
Okay, goodbye.
Yeah. Bye.
You realize that it's 11 o'clock at night here. I have to go to bed now, but
I
it's been
see.
terrific.
See you later,
That's
Dr.
great.
Leslie. Talk to you
Yeah.
again.
See you, Nada.
Bye.
Yes. Bye. Bye.
Bye.
Thank you so much, ma'am.
Thank you so much. Thank you so much. Yes. You can. Yeah, I think it was done nicely. No? Yes. Chalo. So it was good. Anyway, all the speakers were there. They have given very good inputs.
Simple, thank you.
Sabto, thank you. We thank one and all. Every team. Yeah, just keep it at my table. Yeah. So then. Yeah. So we have ended exactly at 6:30. Good, it's good. Because she was there, Nata was there. Yeah, so we thought she may come late, but it's not. So, yes. Good job.